All right, digestive system. So what we're going to be looking at in the digestive system, we're going to look at all the functions related to digestion, obviously. The functions that we'll be looking at will be all contingent based on the organs as we move through the whole entire alimentary canal, which I'll describe on the next couple slides. So everything from ingestion, which is the intake of food, to the secretion of our enzymes, secretion of other fluids such as mucus and saliva. We'll look at digestion, the two different components of digestion where we can undergo mechanical digestion, that is the physical breakdown of food, combine that with the enzymatic reactions from the secreted enzymes in breaking down our food from that perspective. The mixing and propulsion, we'll talk about how do we propel food throughout the entire alimentary canal. And then lastly, how do we deal, or second to last, how do we deal with the absorption of the nutrients from what we ate down to defecation of waste, to remove things that we do not absorb through the alimentary canal. So we're going to be looking from an evolutionary perspective, looking at more of the primitive species. And here you can see our sponge. And the way sponges work essentially is to provide nutrients to these multicellular organisms. And those earlier ways was to take the intake of water. So sponges would basically absorb water. And by absorbing water, I mean it's actually taking in water through these pores. And these pores are able to move through. And any nutrients that are required by the cells of the sponge get absorbed directly into the cell. And anything that is not absorbed will make its way and it's spit right out through this osculum. So this is our primitive, one of our early primitive methods of food absorption. And then with the specialization of the digestive tract, you would see the presence of the alimentary canal. And what exactly is the alimentary canal? And you'll see this you'll see it in this slide. The alimentary canal is everything from the mouth over to the anus. And it is consistent with the external environment. As you saw when we talked about the external environment, whether it's light, sound, you name it, anything that we're interacting with, air, we're taking in things from the external environment and it's going to make its way through the inside of that particular specimen. And what we defecate out is going to still, again, make its way out back into the external environment. So this is our way, same way that sponges were able to take their external environment and move it through the cells, or at least around the cells. We do the same thing with the alimentary canal. And so these species, particularly the nematode, was able to take food, an intake of food through the mouth, and store some of that through the pharynx, and it would just make its way through. And here you see the presence of the intestine, where you have localized absorption of those nutrients, and then it makes its way out its anus. So the issue with the nematode is that it doesn't really have a way of storing food. It's constantly burrowing through and grabbing its nutrients, and it's passing through. Now the earthworm, down this sort of evolutionary line, you can see the earthworm still has that alimentary canal. But what we have, again, is we have our pharynx that assists in part of the digestion, but you see the emergence of a crop and a gizzard. And these allow for enhanced digestion. So you'll see that these specialized organs now secrete enzymes that assist more in the breakdown of those nutrients, so that essentially the earthworm can harvest more nutrients through its environment. And then looking at the salamander, as we again move further down the evolutionary line, you see this presence of the stomach, which allows storage of food. You see the intestine and the emergence of accessory organs, namely the liver as well as the pancreas. And I'll talk about the accessory organs and what that means. Then looking down the digestive tracts, here at the very, in each of these four images, you can see the length and the makeup of the alimentary canal. Everything from the mouth to the esophagus, all the way down to the anus. And here you can see our herbivores, and down here you can see our carnivores. And you can see a clear difference between the two, right? The clear difference is that herbivores have a longer tract. And that should make sense because plants take longer to break down. And so it needs to spend more time through the alimentary canal. It's got to make its way all the way through the small intestine, where we're going to see lengthened absorption. We're going to see the presence of the cecum, which allows the storage after the small intestine, and then through the large intestine. In both our insectivores as well as our carnivores, we don't spend that much time through the small intestine. And that's because breaking down meat takes place much faster than it does our plant material. Or in other words, the diet of carnivores are much more nutrient rich than that of plants. So you can take, for instance, a good example would be those huge whales. I forgot the name of those whales. I'm not much of a biology person, but great whales. No, that's great shark. Killer orca whales. Yeah, the big whale. Yeah, whatever the big whale is that comes in and takes a bunch of plankton in. Those plankton are not very high in calories, so it definitely has to take in quite a bit. And for whatever that name of that whale would spend a long time through the digestive system in order to be able to absorb those nutrients, as opposed to those of carnivores where not much time is allocated to breaking down meat because it breaks down fast and there's a lot of nutrients that can be absorbed in a very calorically dense diet. So as I mentioned, I've been talking about the alimentary canal, again, consistent with that of the external environment. And in humans, you see the alimentary canal travels about 30 feet. It's got a long distance to travel as it makes its way through. The accessory organs, on the other hand, are organs that assist in the alimentary canal. So these would be things like the teeth, the tongue, salivary glands, liver, gallbladder, and the pancreas. The easiest way of thinking about which one would be considered the alimentary canal versus the accessory organs is what food actually travels through. Food travels through the mouth, travels through the esophagus, travels through the stomach, through the small intestine and large intestine. Those are all part of the canal, again, which is consistent with the external environment. Accessory organs, food doesn't travel through the teeth or the liver or the gallbladder or the pancreas. The gallbladder, the liver, and the pancreas dump things into the alimentary canal to assist in digestion as well as absorption. Now, looking at a molecular perspective, we're going to be looking at two terms that we've already seen before: catabolic reactions as well as anabolic reactions. Again, this is a throwback to our biochemical lecture where we saw the breakdown, which is catabolic reactions, taking large molecules and breaking them down to their smaller constituents. This process in our catabolic reactions is basically taking whatever you eat. And whatever you eat, if we're going to take the example of a hamburger, it contains all of the 4 organic compounds, macronutrients, carbohydrates, our proteins, fats, as well as the DNA within those cells. And we're going to break all of those polymers down into their monomers, their individual components. So catabolic reactions are going to be able to release energy. We're releasing the energy that comes from the breakdown of our molecules. This catabolic reaction you saw in cellular respiration. Anabolic reactions are taking those smaller constituents, those monomers, and building larger molecules out of it. So if you've taken that steak and you've broken them down to their amino acids, those amino acids can then again be formed into bigger proteins. We can form them into any globulins, you can form them into hemoglobin, you can form them into any of the proteins that liver produces. You can turn any of those glucose molecules that you've just ingested, that you've broken down through catabolic reactions, and form liver or muscle glycogen for energy storage. So in this process, this is the breakdown of those macronutrients, bringing them to their smaller constituents, and then building back from it. So whenever you hear the concept of you are what you eat, no, you're not. You actually take down whatever you just ate, break them down into smaller things, and you build on top of that. Mechanical digestion. So we're going to start with defining some terms with mechanical versus chemical digestion. What exactly is mechanical digestion? It's basically the physical breakdown of food. How do we break apart that hamburger into smaller pieces? And mechanical digestion first starts off with mastication. That is chewing. what we're doing when we're chewing food. We're using our teeth, the accessory organs, known as our teeth, to physically break that food apart into smaller bites. The process of swallowing, the process of mixing involves whatever you've chewed and swallowed and mixing that with enzymes in the stomach. And now you've got your food mixed together, and what better way to get it to all mixed together through what's a term we're going to call peristalsis, which is going to be muscle movement. Muscle movement in the alimentary canal that allows that mixing of those enzymes, namely the gastric juices that we'll be looking at later, mixing those gastric juices with the food that was chewed and swallowed, allowing us to create this slurry of stuff, and it makes its way through the alimentary canal to further digestion as well as absorption later down the alimentary canal. Now, chemical digestion, on the other hand, is going to involve catabolic reactions. Again, the breakdown, the enzymatic breakdown of our molecules. So in our catabolic reactions, going back to earlier, catabolic reactions involve terms that you'll see later on, hydrolysis, which is the lysing, the break apart of molecules with water. So water molecule comes in and breaks that apart, which you'll see on the back of that original slide. This results in the release of energy. So in other words, this is an exergonic reaction. Now, our endergonic reactions are those for anabolic reactions. So these are endergonic, in which we're going to require energy to build our molecules. So in the case of glycogenesis, that requires ATP. ATP that we were able to harvest from our catabolic reactions. So here you can see catabolic and anabolic reactions together is a whole process of metabolism. For anabolic reactions, this would be called dehydration. Synthesis. In order to synthesize a glycogen molecule, we have to remove water from that process. So going back to that original slide, the breakdown of fats get broken down into fatty acids and glycerol. So in these cases, we're going to be breaking those ester linkages that link the glycerol molecule to those fatty acids. Carbohydrates, any polysaccharides, we're taking those polysaccharides and breaking them down into monosaccharides. Best case example you can see at the bottom is taking table sugar. Table sugar below, which is going to be 1 glucose and 1 fructose saccharide. These two monosaccharides that are linked together by a glycosidic bond get broken up. You have water in that process. Water comes in for hydrolysis, introduce water, and that will break apart that glycosidic bond. Where you originally had an oxygen molecule bound to these two monosaccharides, that gets bound up, you break apart one, and you break apart the other. Again, releasing energy, at the same time requiring a water molecule. So this is something that, in the process of chemical digestion, actually involves water. So you may hear at some point, and if any of you guys ever read any survival stuff, if you're out in a place where you don't have much access to water, but you have food, the process of chemical digestion, of breaking apart those food molecules that you've just taken in, require water, so can contribute to dehydration much faster. The other portion is breaking down our proteins. So again, our polypeptides get broken down into smaller peptides as well as amino acids. We might not be able to break down all these big proteins because again, they might be very long proteins, but we can certainly break them down into smaller peptides that can be absorbed down in the small intestine. Now, the next several slides are going to talk about the cross-section. And I'm going to kind of move through this for the most part so that you guys get the main idea of what's happening, but I'll trust that you guys can go through and read on your own. But through a generic anatomical cross-section of the alimentary canal, again, the canal that food travels through, in the case of the lumen would be the cavity on the inside that food travels through. You can see the different layers, and these layers we've studied quite a bit. You'll see the mucosal layer. The mucosal layer will break down each of the layers. Mucosal layer is the layer that's most exposed to the lumen. You'll see the submucosal layer, which is going to be our connective tissue surrounding. You'll see the muscularis layer, as you can surmise by the name, is going to be the muscular layer. And then the serosa adventitia, the things that are connected to all the surrounding organs. And with that, we'll take a look at the innermost layer, the mucosa. And depending on where we're looking at, we're either going to have non-keratinized stratified squamous epithelium or simple columnar epithelium. I know that sounds like a mouthful, but the first thing we can do is look at the first word. The first word is keratin. Keratin is a protein that's present in hair and nails. So it's a very tough structure. Not something that we would really want in the alimentary canal. So non-keratinized, stratified. What does stratified mean? Multiple layers. Multiple layers of squamous cells, squamous being flat cells. Epithelial cells, which is the characteristic of cells that are meant for protection. So non-keratinized stratified squamous epithelial cells, which you'll be able to see on a later slide, allows for food to travel through areas where we get high traffic. So pharynx, esophagus, as well as the anus. As food travels through, we can slough off the top layers of the epithelial cells. Namely, the top layers, which will get sloughed off, and you still have other flat cells below that still allow for protection. And you'll see that the simple squamous, and these are just a bunch of cells that are all kind of piled up together. As we slough off that top layer, and this is the lumen, and this is our cross section. As that top layer gets sloughed off, you'll see all the basal cells below that are continually dividing to make sure that we serve protection for all the layers underneath, namely the submucosa as well as the muscularis. Now, on the other areas of the alimentary canal, you'll see the simple columnar epithelial cells. Simple meaning one layer thick, columnar just giving the shape, and again, epithelial cells. And in this case, these cells are mainly geared for secretion or absorption, depending on what area of the alimentary canal we're looking at. So again, simple layer, one cell thick, which makes sense for getting things to move across that one cell layer membrane. And where would we see this? We would see this in the stomach as well as the intestines. And then embedded within those epithelial cells, you'll start to see other glandular cells that have exocrine function. These are things that they're going to be secreting into the lumen of that alimentary canal. The layer beneath is just a bunch of connective tissue underneath. Again, we'll look at that later, where we have our capillary network that's going to feed and provide nutrients to the mucosal layer. The muscularis mucosae is a layer within the mucosa itself. This is not the muscularis that you see, which is the third layer. This is the sublayer within the mucosa. And the purpose of that mucosal layer, or the muscularis mucosae, is to provide increased surface area. So you get these folds that are made in the mucosa that allow for nutrient absorption. So what it would look like on that mucosal layer is basically a bunch of folds looking like this. And we'll be able to see those structures as we go through the stomach as well as the small intestine that give us this wrinkled look on the inside. So by increasing the surface area, we increase the amount of contact area that food travels through. We don't want a smooth tube flowing through. We want a bunch of ruffles in there so that it can make its way through and get absorbed as much as possible within the proximity of that lumen, or the mucosal layer. Now within the submucosa, you'll see submucosa again, the lamina propria, which is basically the connective tissue surrounding the mucosal layer, are going to contain your arteries and veins that feed the capillaries embedded within the mucosal layer. So arteries and veins, and naturally, every time when you've done your in anatomy, you've learned when you see arteries and veins, you're also going to see nerves. And that nerve layer is called the Meissner's plexus. And that is going to be the parasympathetic division. These are parasympathetic pathways that come in and innervate the mucosal layer. So this is what we're doing to increase the blood flow to the mucosal layer. So again, parasympathetic pathways, just like you saw in the test. Parasympathetic pathways that allow vasodilation to increase blood flow to the mucosal layer. Now the muscularis layer, again, the outer layer on here, is basically going to consist of skeletal muscle, or smooth muscle. And again, depending on where we are in the alimentary canal, you're going to either see voluntary actions or involuntary actions. Voluntary include the act of swallowing, control of defecation, where we see skeletal muscle that'll control either input or the output of food through the alimentary canal. Smooth muscle is going to involve, areas with smooth muscle are going to involve autonomic regulation. So these are going to be areas that are going to contract on their own. Involuntary control of those areas. And typically in the muscularis layer, you're going to see two different layers. And if I can just draw just a generic tube through, you're going to see these circular layers. You're going to have a circular layer, and then you're going to have a longitudinal sheet that runs along the length of the canal itself. And the purpose of this is to assist in the squeezing that takes place. You're going to squeeze segment by segment to be able to propel food down the alimentary canal. So that's going to be consistent. The intercircular muscles are going to allow contraction within that particular unit. And then the longitudinal sheets are going to create these wave-like patterns that will allow the propulsion of food through down the length of that canal. The myenteric nerve plexus. That basically just allows innervation of the mucosal layer for the production of our chemical enzymes and so forth. So myenteric nerve plexus are things that you're going to be able to see in the stomach that allow the mucosal layer to do what it's supposed to do. Serosa and adventitia, again, the outer layer. This is going to be the outer layer that wraps around. Namely, it's going to contain serous fluid. Serous fluid is basically a lubricant. It allows for the slipperiness. Now imagine, you've just eaten something and you've got all this muscle movement taking place in the alimentary canal. So all the surrounding tissues will want to be surrounded by serous fluid to allow that slipperiness. You don't want friction happening along the organs. You want that fluid to allow those tissues to slip and slide around each other. That's all I really want to mention on that side. The next couple slides are going to talk about the peritoneum. This I don't need you guys to really focus too much on. Basically, all this is the protection of the GI tract. And in these cases, you're going to get these fatty layers that come in and protect the anterior of the body. As you know, we've got the ribs that help protect the lungs, the thoracic cavity, but nothing really protects the abdomen. And that's where we get the peritoneum that comes in and allows for the protection of that. And there's numerous folds, which I won't really get into. The biggest one of them is called the greater omentum. And the greater omentum is basically a fatty apron. It's the one that attaches right underneath the stomach and folds over down towards the bottom of the small intestines and wraps right back up and attaches to the end, attaches underneath. So here you can see the greater omentum that's kind of lifted up. This, again, the fatty apron is typically where you would see what the so-called beer belly. The beer belly is where you get most of the fat deposition that takes place in males. Okay. Next, looking at the phases of digestion is to look at everything that takes place from the mouth all the way down to the anus. And the phases of digestion, well, technically we're not doing anything, we're not digesting in the anus, so it's all up down to the very end where we see the small intestines. So we're going to start from the head down to the small intestines. And namely, these three different phases are named based on the organs that it contains. Cephalic, again, meaning the head. Gastric phase involving the stomach. And the intestinal phase involving the small intestines. So each of these areas are going to contribute to the different phases of digestion that takes place. And first and foremost, you can see the small intestine also involved in digestion. This is something that I've seen that people sometimes don't really think about, is that it doesn't end at the stomach. We're going to do more digestion within the small intestine. And this is actually the longest pathway in the alimentary canal, where the small intestine is going to involve digestion as well as the other function for absorption. But right now we're just talking about digestion. So what happens in the head, the cephalic phase? this is where we're going to start to involve sensory reception. We're going to receive sensory information. What information are we receiving? The smell of food, the taste of food, as well as the texture of food. So all that sensory input is going to lead into the central nervous system, and it's going to start triggering the other organs in place. Okay? So if any of you guys, I don't know if you guys eat dinner that late by the time you get home. If you're eating something, you might notice that once you start eating, you might even look at food, and you notice that your stomach starts grumbling. Why is that? Well, that's the process. When you start seeing food, you start smelling it, that's trying to prep the other organs. It's going to say, you're about to start eating some food, so start ramping up the stomach, start ramping up the small intestine, start producing your enzymes, because there's some things that we're going to start getting the intake of food taking place. Best case example is being exposed to food and salivating. The process of the production of saliva is an autonomic response. Okay? Again, it is a parasympathetic activity, as you mentioned, SLUDD. Salivation is something that is parasympathetic. Again, a response due to that sensory information into the central nervous system. We'll learn about what other forms of digestion are taking place in the mouth. The gastric phase, as we move from the mouth down to the stomach, we're going to learn about communication. Communication from the central nervous system, our neural regulation, as well as the hormonal regulation that takes place to supplement central nervous pathways. So neural regulation, this involves the input of information. These involve the output of information. There's still some CNS sensory information taking place within the stomach, which we'll learn later on, but that is also going to trigger the ways of releasing digestive enzymes, as well as the hormones that are involved. So neural regulation involves the production of our enzymes, as well as our other digestive juices, as well as the hormone regulation, which is going to involve hormones that are going to allow communication with other organs downstream. And this, which we'll talk about later on, is called gastrin. The last phase would be the intestinal phase. And in the intestinal phase, we're moving from the stomach over to the intestines. And here you're going to learn about the two enzymes, cholecystokinin as well as secretin. Now, before I get into cholecystokinin as well as secretin, I want to talk about the pH, which we'll learn much later on. The pH of saliva, again, which you'll learn later on, is about 6.5. So it's easy to say that this is a chemically neutral or a pH neutral environment. By the time we make our way into the stomach, we're going to have a very highly acidic environment where the pH is around 2. So we have a lot of hydrogen ions in this process. And we're going to learn about how we produce the acids present in the stomach. And then when we make our way into the small intestine, we're going to be back at a pH of 7. So imagine the fluid that flows from the stomach that's going to have a pH of 2 is going to get dumped into the small intestine. And we're certainly going to need some form of a buffer to prevent the pH changes, to bring it back, well, not prevent the pH changes, but to bring it back to a neutral pH. And that molecule is bicarbonate. In the same pathway that you learned about the production of bicarbonate in our red blood cells, namely through the enzyme called carbonic anhydrase, is involved, the same enzyme is involved in the production of bicarbonate. So bicarbonate in this case is going to serve to buffer the acidic chyme, which we'll learn later on, the acidic fluid that makes its way into the small intestine. At the same time, we do have accessory organs. Our accessory organs are going to be our pancreas, which is going to produce our bile, and the gallbladder that's going to be involved in the storage of bile. So those enzymes, which we'll talk about later, okay, are going to be involved in the digestion of fats. So if these organs are not actually part of the alimentary canal, and let me just draw it off key somewhere here, and I know this is not the right place for the pancreas, but as food travels through the small intestine, we need to have a hormone that's going to come in and signal the pancreas to start dumping its bile back into the small intestine. So that's where CCK comes in, cholecystokinin. And you'll learn about the origin of that name later on so that it's easier to memorize. So that pretty much sums up digestion. And we're going to learn about the pathways involved, where gastrin is going to be involved. That's going to assist in the production of our gastric juices. We'll learn about the regulation that takes place. When do we produce gastrin? And when do we stop producing gastrin? And with that, we'll take a look at the pathway of food. We move everywhere from the mouth, the pharynx, esophagus, stomach, small intestine. Small intestine is going to be divided into the duodenum, jejunum, as well as the ileum. Okay? Then we move to the large intestine, to the colon, to the rectum, to the anus. Self-explanatory. Now with that, we're going to start describing all the organs as well as the accessory organs through the alimentary canal. And as we know, alimentary canal starts at the mouth. We start at the mouth, also known as the buccal cavity, and again formed by the cheeks, as well as the hard palate, which is more anterior, and the soft palate, which is much more posterior in the back of the mouth, as well as the tongue. This cavity itself is the alimentary canal, the mouth being the alimentary canal, the cheeks, the soft palate, and the tongue being accessory organs that contribute to the cavity itself, and of which we're going to undergo two different forms of digestion, of which we illustrated earlier. Mechanical digestion, which is involving the process of mastication. We're chewing. We take food, we put it into our mouth, we use the teeth to bite the food into smaller pieces, and we're mashing that food. Not only are we biting the food, we're using our teeth to mash the saliva in with that food that we've just ingested. And we use the muscles in the lips and the cheeks to focus that food within the teeth. So think about what your tongue, as well as your cheeks, to focus that food directly onto the teeth to help mash that mashing to take place. So in that process, we're going to create the first step of what we call our food, is that bolus of food, which is going to be what we've ingested. Combine that with saliva. We'll talk about what's in saliva on a later slide. Chemical digestion are going to involve the enzymes in this process. So we'll learn about what's composed in saliva, of which you're going to see most of it being water, about 99.5% water. Dissolved ions are going to be involved in the enzymes itself. Immunoglobulins, which are all involved in the immune system. These are things to help prevent any viruses or disease that come in, not viruses or bacteria, that these immunoglobulins come in and attack to prevent it from entering into our body. Lysozymes are going to be, again, a bacteriolytic enzyme, basically breaking down bacteria for things that don't belong. And then the first of our enzymes are called salivary amylase. And amylase is going to be the first of our enzymes, a digestive enzyme for starch. It's what we first do to break down starch. What else is also present in saliva? We actually also have a very weak painkiller, and I keep forgetting the name of it. It's like oro something. It's a painkiller that's actually present in our saliva in very, very minute amounts. As well, combine that with immunoglobulins, sort of explains why dogs, or even cats, why they lick themselves. If you ever watch a dog lick their wound, why are they licking their wound? They're not doing it because it tastes good. They're doing it because their saliva, by instinct, contains that painkiller. It's a very weak painkiller that comes in. And then the second thing that it also does, it introduces immunoglobulins that come in and also attack the bacteria that might be present in that wound. And the other thing that they're also doing by licking is actually removing any sort of particulate matter that might be present in the wound itself. So there is an instinctual need for animals to lick their wounds. Well, why do we not lick our wounds? Well, obviously we've got better technology than licking our own wounds. Number two, it's kind of gross. I don't know about tasting your own blood, which I don't know, I might have an aversion to. But not only that, we also have a lot of bacteria that's also present too. So you have heard, if you ever look up cases of people that have been able to heal wounds, some of the earlier cases they've been able to use saliva for that, but also at the same time they've also had cases where you've had people get amputated, get limbs amputated because of the introduction of a bacteria that was only present in the mouth as well. So don't lick your wounds. Rely on better medicine than that. Salivary glands. You'll see that we have 3 major salivary glands located in different areas of the mouth. And these salivary glands are tasked for the production of saliva. So we've got 3 glands, parotid, submandibular, and the sublingual glands. Parotid being present up at the top sides, lateral sides of the mouth. You'll see the submandibular, which is well below the mouth. And then you've got the sublingual, which is below the tongue. How do you know these areas? You ever bite into anything sour? You bite into a lemon, you might notice that you'll produce a lot of saliva. All of a sudden your cheeks get super watery. Why is that? You've activated your parotid glands. The parotid glands are secreting mucus. Why is that? Why does it do that? Well, think about what happens when you're biting into a lemon. It's something acidic. The pH of your saliva, as we mentioned earlier, is 6.5. And anything that comes in and lowers the pH, you're going to have an autonomic reaction where the parotid gland will dump in a bunch of watery saliva to help bring the pH back up towards 6.5. So that is, in essence, a form of negative feedback. What starts off with a drop in pH results in a bunch of water being dumped in to raise pH back up. What's characteristic of the parotid gland, as I mentioned earlier, this creates more of a watery saliva, where most of it's going to be water. The other portion of saliva consists of water plus mucus. And mucus basically is a protein called mucin plus water. So introduce this mucus. Where does mucus come from? Mucus is present in saliva, but our submandibular and even more so our sublingual glands produce more of a mucousy saliva. So this sublingual gland typically is a thicker mucus, as opposed to the parotid, which is going to produce more of a watery mucus. More to come on that on a couple slides over, which I'll talk about. Other areas that also produce saliva, the lips produce a little bit of it, the cheeks, palate at the very top, as well as the tongue itself have very minor salivary glands. But the biggest contributor of those are our 3 at the very top. How much saliva do we end up producing? We produce about 1 to about 1.5 liters of saliva per day. That's a lot of spit. What function does it serve? Coats the oral mucosa. Keeps the mouth moist. And why is that? Well, think about in our sensory lecture. Whenever we break apart food, we're releasing some of the tastings that come off food, and that dissolves in that watery environment. So we require saliva in order to dissolve those tastings so that they can make their way into the gustatory taste buds. And eventually those gustatory receptors will be able to pick up those flavors and send that over to the central nervous system. As I mentioned earlier, it's an acid buffer. So it does contain bicarbonate, so if you ever do end up eating anything sour, that comes in and that bicarbonate comes in and buffers that acid. Another thing too is soda. So those that drink soda, and I know I drink quite a bit of it, even diet though, there's studies that show that diet soda isn't really that great for you. But soda in general contains citric acid, and citric acid is an acid, and actually will lower the pH of your mouth. And so what you'll notice about soda consumption is that you produce a lot of saliva. And what comes in saliva? Within that saliva, you've got a bunch of ions in there that can start to form calcium deposits. And saliva by itself actually has something that prevents the formation of those calcium deposits. But nonetheless, those that continually drink soda will actually produce more and more saliva. And eventually when you exhaust the buffering capacity of saliva, that's where the pH of your mouth starts to drop and can cause the breakdown of an enamel. Any of you guys know anybody, or even you guys drink like Mountain Dew or any of the citrusy type of sodas? Nobody? like, I don't know, squirt, cactus coolers, and I don't even know about cactus cooler, 7-Up, for instance, Mountain Dew. Those actually have more citric acid, which is actually bad for you. And what's even worse are those that drink warm Mountain Dew. If you drink, and it doesn't have to be warm, it's just not super cold. And then even worse than that are people that sip on it throughout the day. So you're constantly, those that do that, and if you know anybody that does that, take a look at their teeth, and they've worn away the outer layer. You're taking in an acid, a warm acid. Again, what did temperature do? It increases the kinetic energy. So you've got an acid that's coming away, chipping away at that outer layer in the teeth, which we'll look at a little bit later, and eventually will cause exposure of the inner layers of teeth and teeth rotting. I know at some point, parents would have told you don't drink soda because it'll rot your teeth. They're actually right on that one. I know they spit a lot of like old wives' tales, but it actually will do that. Other things too, antimicrobial, which we mentioned earlier, tissue repair, which we mentioned what dogs do. Again, the presence of painkiller as well as other formation of, it was called, not anything you need to know, salivary. What that does, it allows to allow the speed up of tissue repair. So it enhances tissue repair. It's a form of growth factor, which comes in and contributes to the repair of any damaged tissues. Also in saliva, contributes to digestion, called amylase. Amylase is active, which we'll see later on, is active in a pH of about 6 to 7. We'll learn later what's going to inactivate amylase. Okay, taste, which we described earlier. Now, looking at salivary regulation, I mentioned that the production of saliva is a parasympathetic reaction. But technically, it's actually also sympathetic as well. So it's not only parasympathetic, it's also sympathetic. So I lied to you a little bit. But in parasympathetic stimulation, you produce watery saliva. So the salivary pathways from the brainstem to the medulla, all the way out to the salivary glands, produces a watery saliva. And that makes sense because what we're doing is when we introduce food into our mouth, we want to be able to mash a bunch of that food with that watery saliva. And that's going to carry all the digestive enzymes and so forth. So all these things right here, all the sensory information, stimulate the production of saliva. The touch of food, any texture that's felt by the tongue, the smell of food, the taste of food, and other psychological factors. What psychological factors are? Just thinking about food stimulates the production of saliva. If you ever think about the old experiments where we had Pavlov's dogs, and those dogs got exposed to meat powder, those dogs would salivate. It was a completely autonomic reaction. It's just they smelled food and all of a sudden they salivate. You might be doing that too if you're exposed to food as well. All of a sudden you notice that your mouth starts to get pretty filled with saliva. And again, all that is to contribute to the digestion that takes place. So what did I mean by sympathetic pathways creating saliva as well? Well, the makeup of saliva in sympathetic pathways produces much more mucus than we produce water. So in essence, the saliva becomes much thicker. Hence the sort of dry mouth that you're experiencing whenever you're under any sympathetic, whenever you're undergoing any sympathetic reaction. So if this has ever happened to you where you've had like an absolute fright of something, all of a sudden your lips get dry, your mouth gets dry, well technically what's happening is you're still producing saliva, but it's much more mucus, which gives you the feeling that your mouth is dry, when in essence, you're just producing something that absorbs that water, and that would be your mucus. And why do we do that? Well, in our sympathetic pathways, we know we're breathing a lot more. It's going to cause increased respiration and increased ventilation. And that means more air is going to be traveling through the mouth and the pharynx as well. So we know that mucus, being sticky as it is, allows the trapping of particulates that might be embedded in air. So any dust particles, any pollen are going to stick to the mucus and not make its way into the lungs. Hence why we need a thicker mucus for that process. Now the tongue, you'll see, as I mentioned earlier, the tongue is tasked to keep food right by, in between the teeth. It's going to be involved, at least in the mechanical digestion. The muscular action that takes place, you have the tongue, which is attached, allows moving the tongue side to side, and the intrinsic muscles within the tongue allow for the shaping of the tongue itself, so to produce the sounds that we do during speech. And then also the tongue involved in swallowing. I'm not going to spend too much time on this because that is fairly obvious. The teeth are the mouth itself. You know what a mouth looks like. You'll see all the different structures contained within. I don't really talk so much about it. The teeth itself, I don't really talk about much either. Why? Because you know teeth. For the most part, you'll see the outer layer, the inner layer, the middle layer. I don't really talk too much about it. Other than the enamel will be the outer layer, the dentin will be the middle layer, which you'll see on the next slide over. The pulp inside the tooth itself, it happens to be hollow. It contains the arteries, arteries, veins, and the nerves. You'll see the crown of the tooth, which is the top layer that's above the gum line. You'll see the neck, and then the root, which is going to be the root tips. The mouth, we have 32 adult teeth, 20 baby teeth. One thing also too, is characteristic of omnivores. You have your incisors, which are sharp, meant for biting. So if you're biting into a carrot, biting into an apple, we're using the incisors to basically take a chunk out. And then you get the molars in the back. The molars have a flat surface that allow for grinding. And again, to mechanically break down our food into smaller parts, but also to allow mashing of that food with saliva as well. All right, that's it with the mouth in terms of the structures. Mastication, I won't go into more detail because I've already talked about that. This is where we're breaking down our food to create a bolus. Now let's talk about chemical digestion. We've talked about salivary amylase, which breaks down starch. So this is the first of our active enzymes. It's active in at a pH about 6.5 to 7. And by the time that amylase makes its way down into the stomach, it's deactivated or inactivated due to the acidic environment of the stomach. So thinking about an enzyme, let me just draw kind of just a random structure there. If this is what amylase looks like, and I have no idea what amylase looks like, but if amylase looks like this, what happens in the case of an acidic environment? We have an increased amount of hydrogen ions. Those hydrogen ions come in and interfere with the secondary and tertiary structures, causing denaturing of the enzyme. So now all of a sudden we get our enzyme to look like, I don't know, something like a circle or something like that, I don't know, whatever it looks like, which interferes with the active site in which starch binds to amylase. So that denaturing of the enzyme that takes place due to the acidic environment takes place in the stomach. So it's active in the mouth and inactivated into the stomach. Now, on the other hand, we have actually another enzyme, and that enzyme is called lingual lipase. So this is produced by the tongue itself. This lipase, again, looking at all of our enzymes, end in the term ACE. A-S-E. Lingual lipase is for the breakdown, salivary amylase is for the breakdown of starch, lipase is for the breakdown of fats. But it's not necessarily active in the mouth. Why? It requires a low pH. So technically, it's denatured in the mouth, and by the time it makes its way to an acidic environment, this is where lipase unravels and opens up that active site. So denaturing in this place isn't necessarily due to the hydrogen ions. It's really due to the chemical and the pH that allows for the optimal operation of that particular molecule. So in this case, amylase is active in a neutral pH. Lipase requires the gastric juices in order to become active and start breaking down fats. So in sum, where do we start digesting, at least chemically digesting starches? That takes place in the mouth. Yes?
So pretty much there's no starch breakdown in the stomach, but
There's still, it just starts in the mouth.
Oh, okay.
What's relevant with this? Something that I actually saw was really interesting is those that practice Ramadan cannot typically are told not to. I don't think it's the act of swallowing that's not allowed, I believe. So you're not allowed to swallow food or anything like that. But the loophole there is that you're allowed to kind of swish stuff around your mouth. So if you were to, I don't think you're allowed to drink water, but you're allowed to like swish water into your mouth, but as long as you spit it out. Something that was a poster that I went to a convention a long time ago was looking at ways for people, especially marathon runners. Marathon runners that practiced Ramadan. Okay, marathon runners. If you ever look at marathon runners, they take in gels, right? So after, did anybody ever run a marathon here? Nobody? No? Okay. Well, anyway, not like I ever ran a marathon. But those that run marathons will eventually hit a wall, and what this so-called wall is actually is a physiological point at which you have low blood sugar. And in order to raise your blood sugar, best way is to take in some sort of carbohydrate gel. Either you're taking in some sugar substitute of some, not sugar substitute, but you're taking in sugar some way or another to raise your blood sugar back up. But those that practice during Ramadan, during this period of fasting, are not allowed to take in food. So what they learn in this, it was a poster or it was a talk that I attended. One of the things that people that were practicing Muslim during Ramadan was that they could take in glucose. They can take in any sugary substitute, and they can swish it around their mouth. And it was okay because salivary amylase would break apart those starches, and you still absorb through the mouth. Why? Because you've got capillaries right along there. And you do absorb a very small amount, but that small amount was sufficient to keep these marathon runners running without technically ever swallowing food. So you'd ever watch anybody that was practicing during that time, they could swish sugary stuff around their mouth and spit it right back out and still get the glucose they need to raise their blood sugar back up. Process of swallowing, we're going to move our way down the pharynx. And as you saw earlier, the questions that you guys were able to answer earlier on the test about the epiglottis. Epiglottis is going to be involved, which I'll talk about in a later slide. But basically, the pharynx is going to be the pathway, the next pathway for swallowing the food from the mouth. So mouth down to the pharynx, we'll see a pathway that's involved, or a process that's involved in preventing food from entering the respiratory tract. When we're swallowing, we're going to break this down to 3 different phases: the voluntary, pharyngeal, and esophageal phases. In this process of swallowing, the technical term being deglutition, you'll see the process that's taking place is the bolus of food that's present in the mouth. And the sphincter that's going to be involved, which you'll see later on, is the upper esophageal sphincter. When we talk about the esophagus, we'll talk about these. And the esophageal sphincter is right up above of the esophagus. The bolus of food that's present, and in the act of swallowing or deglutition, the first process is the soft palate, which is the palate behind the hard palate. So this particular structure right here will move upwards to block the nasal passage. So that way any air that takes place during swallowing, think about it, when you're swallowing, you're not actually inhaling air because we've occluded the nasopharynx. We can't allow any air to travel during that time. At the same time, you'll see the epiglottis. The epiglottis that's present right up here folds over and blocks the trachea to prevent food from traveling down the trachea. So this process, in order to move the soft palate as well as the epiglottis, is autonomically regulated. This is just a reflex that takes place to prevent food from traveling superiorly and to allow it to travel down the esophagus. Once we start to swallow, this opens up the upper esophageal sphincter, and then once we finish swallowing, you'll see everything moves back to its original configuration. The soft palate moves back down, the epiglottis moves back up, and the sphincter closes right back up to prevent anything moving down the esophagus that shouldn't be. So breaking down these different phases, the very first phase, the voluntary phase, the act of swallowing is voluntary control. You control when you initially swallow. And in that process, what your tongue is doing is forcing that food. See how the tongue changes the shape, and it allows the food, the bolus of food to come in by the act of swallowing is going to come in and force that food to the back of the back of the throat. So this is the changing shape of the tongue, and it pushes the food, allows it to slide. That bolus of food slides back down, pinches right off at the top. Once that makes its way down to the pharynx, now it's completely involuntary. Everything from this point is autonomic. As the bolus travels down, we do have impulses from the deglutition center in the medulla. So medulla is aware that you're swallowing something in, and that's going to move the soft palate as well as the epiglottis. Now the esophageal stage is now moving its way down to the esophagus. Once the bolus enters the esophagus, you're going to see peristalsis. And again, peristalsis, if I can draw the layer of let me just draw it vertically. Here's our esophagus. We've got our circular layers, and then we have our longitudinal layers. And those longitudinal layers, combine that with our circular layers, allow the squeezing as that bolus travels. So you get that bolus of food down here, you get the squeezing that takes place that allow it to force down to the next segment. And then again, the longitudinal waves that come in and force that bolus of food down. So how long does it take for food to travel through? It takes actually fairly quickly. It's a couple seconds for that bolus of food to make its way down. Water actually moves down fairly quickly. It takes about a second to make its way down into the stomach. So if you drink water very quickly, you can feel it moving its way through and it just enters your stomach very quickly. And that's because there's not really much peristalsis that's needed to assist water to move down. It's already slippery as is. It doesn't rely as much on the peristaltic waves. Also think about what happens in space. Those that are in space where there is no gravity, how do they get food to make its way from the mouth down into the stomach? Again, peristaltic waves, which is basically just all the contractions that take place to move food down. It's like taking a toothpaste tube, squeezing it, and wringing it down towards the neck of the tube. That's basically what's happening in peristalsis. Now, the rest from the esophagus, as I mentioned, moving food down the esophagus. I really have to talk more about that because that's self-explanatory. Looking at the esophagus, as I mentioned, our histological slide, non-keratinized stratified squamous epithelium. You can see that right up at the very top. So flat cells, all stacked up, multi-layers thick. And looking at the muscularis, the layer, you have the mucosa, you have the submucosa, and the muscularis layer. Down the length of the esophagus, you'll see the first third being skeletal. You'll see the next layer being a mix of the two. And the bottom layer being smooth muscle. So there's a transition as we move down the length of the esophagus of which layers are smooth versus skeletal. The different sphincters in the esophagus, the upper esophageal sphincter, which is involved in the right during swallowing. and then the lower esophageal sphincter, which is located below. The esophageal sphincter, the lower esophageal sphincter is the one that separates the stomach from that of the esophagus. And that area is right over here. This allows that bolus of food to make its way through and to make its way into the stomach. This area, though, is a common area for what's called GERD. GERD is, it's already written down for you below, gastroesophageal reflux disease, where our stomach has a pH of 2, and our esophagus is probably around a pH of about 6. So what happens with incompetence of this sphincter is that if this sphincter cannot tightly close, that causes any of the stomach contents, namely the acids and gastric juices, that can start to make its way and leak through that esophageal sphincter and can come in and irritate the sides of the esophagus. So that irritation that takes place causes the inflammation of that area. GERD becomes an issue. It's a gastroesophageal reflux disease where you get those acidic stuff making its way up. Now, that inflammation that takes place is what is an issue with people that have heartburn. Another thing that's also prominent amongst those that have respiratory diseases typically also will have GERD. So any of you guys that see pulmonary patients typically will also be prescribed proton inhibitors. Proton inhibitors that would inhibit the production of protons. What protons are we talking about? Hydrogen protons, hydrogen H+. The things that contribute to the acidic nature. So if you can limit the production of acids, you can reduce the amount of inflammation due to those acids in the esophagus. So these typical drugs like omeprazole or pantoprazole typically will end in P-R-A-Z-O-L-E. These types of drugs are called proton inhibitors. So as I mentioned earlier, anybody that presents with respiratory diseases typically might actually have respiratory diseases due to GERD, right? So in those cases, stomach acids may make their way back up. They can make their way back up, and they can be aspirated, meaning they can end up inhaling some of that gastric juices into the lungs, causing inflammation of the airways. So typically anybody that presents, I don't say typically, but anybody that presents with any pulmonary diseases might also have these as well. So if you're ever looking at their medication history, you might see that on there as well. So this video just shows you what stomach is taking place. Glad you can't hear it. It's probably the loudest I can get it. That's why. That's not why.
Swallowing is simple. Conditions reflexes. Food entry. Respiratory passages. Nuclear contracts, which blocks the nasal passages from the lungs. Laryngeal muscles contract, closing the lines at the top of the trachea, tightening the lines of both. The upper glottis swings down on the bones of the lungs. All areas blocked off. Respiration is temporarily made. As the upper esophageal sphincter rises, usual contractions drive the bolus into the esophagus. The oral pharyngeal stage is done, and then resumes. During the esophageal stage, the primary wave of peristalsis, initiated by the swallowing sphincter, pushes the bolus into the
So
esophagus.
here you can see peristaltic waves,
Travels through the esophagus.
either circular muscles as well as the longitudinal muscles, lower esophageal sphincter that comes in and dumps the food in the contents of the stomach.
Goes through the esophagus. Secondary peristaltic.
As I mentioned earlier, food takes about a couple seconds, about 4 to 5 seconds for food to make its way through, depending on how dry something is. So the presence of the stomach, when we look at the shape of the organ itself, it's J-shaped, simply because it's the shape of a J. And what's particular about the stomach is the presence, or actually let's even talk about the function of the stomach. What exactly is the stomach for? It's for the storage of food. Whatever we've just consumed and ingested and has been propulsed down the esophagus has made its way into the stomach, where we've got a container, which is actually a fairly small container. It's about 50 milliliters in size. It's about 2 ounces worth. It's about 2 shots worth it, and fairly compact. And it allows for distension, about 15 times its size. It can now expand quite a bit. And in the stomach, what you'll notice about the mucosae is the muscularis mucosae on the inside, that the layer within the mucosae are quite folded. And that folds that are present are called rugae. So that allows that distension from, that allows the expansion of the stomach. So in essence, a closed stomach or an empty stomach is quite wrinkled. And in order for that to distend or expand, that's where the rugae allows the expansion of it. So if you were to take a look at, like, for instance, I don't know if you guys ever like look at a pug's face and see how like how wrinkled it is, and you like stretch it out. That's exactly what the stomach looks like. Another thing to think about, this is something that I'm quite guilty of, especially during Thanksgiving, is the effect of ventilation on stomach positions. If you ever notice, after eating a fairly heavy meal,
Can't breathe.
you can't breathe.
Yeah.
Why is that?
Because you eat too much.
Of course you eat too much, that's partly it. But looking at the position of where the stomach is, the esophagus, right in the diaphragm, you have a little area called the esophageal hiatus. It's basically nestled in where the esophagus can travel through the hiatus and to make its way into the stomach. So above the stomach, just superior to the stomach, is the diaphragm. And what did we learn about inspiration? Inspiration involves the contraction of the diaphragm. So it moves posteriorly. So contraction of the diaphragm pushes down on the stomach. Now, when the stomach fills up, you're going to increase the amount of abdominal pressure. At the same time, with inspiration, you're trying to push down on something that's already quite a bit of pressure. So this issue isn't so much of an issue in healthy patients, but it's a particular issue for those that have respiratory issues. They mentioned don't have heavy meals because it's going to be harder for you to breathe. Now, looking at the rugae, you'll see this is an endoscope. And in this endoscope, they're basically going into the stomach itself. You can see the presence of the rugae. If I can just play that for you. And they're making their way in. You can see all the wrinkles. Great. Now, what are the functions of it? Like I said, a food reservoir, about 50 milliliters when empty, and about 1 liter at capacity. So you can see how much we can expand that stomach. What do we do when we're with that food reservoir? We have, again, a bunch of gastric juices, which is going to be a bunch of water, at the same time also a bunch of enzymes, and a lot of protons, our hydrogen ions, that are all going to mix together. So we get the boluses of food that are dumping into the stomach, and then we're going to allow this mixing that takes place of the gastric juices along with the boluses of food. So here we're going to involve, again, peristaltic movements within the muscularis layer that creates these mixing waves. Basically, this is your stomach sort of churning every 15 to 25 seconds to allow mixing of the gastric juices with the food you've just ingested. So in this process where you get this mixing, you're mixing the enzymes to allow chemical digestion to take place, and then you get the physical, the mechanical digestion of that mixing of the food through the peristalsis, and the solution that we end up with is called chyme. So chyme is gastric juice plus the food, plus the ingested boluses. With that, in this process that we'll learn about, what's causing the stomach to expand with the filling of that food, just like we learned in the herring brewer reflex in in the lungs, we also have stretch receptors in the stomach. So as the stomach stretches, you're going to learn that the stretch receptors are going to pick that up, and they're also going to start circulating hormones, which you'll learn later on. That's going to lend itself back to the central nervous system to tell you you're full. So those are the feelings of what's called satiety. Satiety is the feeling of fullness. And these are going to produce signals to stop eating. What's particular about the muscularis layer on the stomach is that I've talked about two different layers. I've talked about the circular layer, and I've also talked about the longitudinal layer. There's actually a third layer called the oblique layer, and that's present in the stomach as well, in the muscularis layer, to allow additional digestion from to allow digestion to take place. And so you can see that present. You'll see the outer layer, which is the longitudinal layer, and that again runs along the entire length of the alimentary canal. You'll see the circular layer, which is just wrapping around the tube, and then the oblique layer that wraps diagonally. And that contributes to those mixing waves within the stomach. The different regions of the stomach, you'll see the cardia, which is up at the towards by the esophageal sphincter, again, an area that's prone to inflammation, close to the esophageal sphincter. You get the fundus, which is the very top. Typically the fundus area is more the reservoir area that fills up. The area that actually does fill up quite a bit would be the pylorus, which is by the pyloric sphincter below where the small intestine starts. So pylorus, and then the body, which is going to be right up above. So as we fill up with food right down, as the stomach starts to fill up with chyme, you'll see most of it is within the pylorus itself. What's technically happening in the stomach is to squeeze small amounts of chyme into the small intestine. So there's some squirting going on where the contractions of the pylorus is going to squeeze little bits in conjunction with the pyloric sphincter, which is going to allow little bits of chyme to make its way down the small intestine. So this process starts the process at which we're slowing down through the small intestine. And again, a pH of 2 and a pH of 7. And we're going to learn where the pancreas is going to be involved in dumping a bunch of secretin to buffer that chyme that makes its way in. More to come on that on Tuesday. So in that process, that emptying is called gastric emptying. And we'll talk about the rates of gastric emptying later on. Take a histological slide. Again, you'll see the presence of, again, the mucosal layer, the submucosa, which is going to contain the arteries and the veins and the nerves, and then the three different layers of the muscularis. Again, longitudinal being the outermost. You'll see the circularis, the circular muscles, as well as the obliques. Now, looking at the mucosal layer, again, the presence of rugae, the wrinkles. Within the rugae, you'll see the reason for those wrinkles are going to create these little pockets. And these pockets within the rugae are going to be the presence of exocrine or secretory cells. So not only exocrine, but also endocrine cells. So within these pockets, you're going to have these specialized epithelial cells that are going to have very specific functions. And those functions, which you'll learn later on, are the production of our acids, as well as mucus. These little gastric pits at the very bottom are going to have the gastric glands. So looking at these specialized cells, we have both exocrine and endocrine. First look at our exocrine cells. Why are they called exocrine? Because exocrine, again, remember, is dumping into the external environment. And given that the lumen of the stomach is the external environment, we're dumping things from within these cells into the gastric pits, which will make its way out into the lumen. So they are exocrine in nature. And the three that we'll talk about are mucous neck cells, which are prominent towards more towards the top of the gastric pit. You also have surface mucus cells, which are up at the very top. The mucous neck cells, we have our parietal cells, and then we have our chief cells. parietal cells are going to be the ones that produce what's called an intrinsic factor. And that intrinsic factor we'll talk a little bit about later on. But the main idea behind parietal cells is that they produce hydrochloric acid. HCl. So combine those protons with chloride anions, and you get hydrochloric acid. Now, chief cells are going to be secreting a protease. ACE being an enzyme, protease referring to an enzyme that breaks down protein. And the first one that's called is pepsinogen. Pepsinogen, which we'll learn in just a little bit. Pepsinogen is the inactivated form. And by the time it makes its way into the low pH, a pH of 2, it becomes pepsin. So we go from the inactivated term, so ogen, anything that ends in ogen is the inactivated. Okay. Inactivated, and then active enzyme will be pepsin. Okay. If you remember angiotensinogen and angiotensin. Again, angiotensinogen being the inactive form, angiotensin being the active form. Same thing here. One way of remembering the chief cells for the production of the protein enzyme pepsinogen is something that's taught in med school. Is chief cells looks like chef cells, and chef cuts the meat. The other thing that's also produced by chief cells are gastric lipase. So this is different than the lingual lipase. Lingual lipase is produced by the tongue. Gastric lipase is produced by the chief cells. And both lipases serve the function of breaking apart fats. So the two enzymes, the two lipases, lingual lipase as well as gastric lipase, work to break down fats in the stomach. Again, due to the pH of 2. The other endocrine cells would be called G cells. So this is the, and not a term you really need to know, but enteroendocrine. So an enteroendocrinologist focus specifically on the endocrine cells within the stomach. And this are produced by G cells that produce gastrin. And gastrins are going to be the cells that we'll learn later on are going to promote the production of hydrochloric acid as well as the other exocrine cells. So in essence, these cells trigger localized production of hydrochloric acid by the parietal cells as well as pepsinogen and gastric lipase by the chief cells. They're the ones communicating to those exocrine cells saying, start producing stuff. You've got food entering the stomach. I need you guys to start producing a bunch of hydrochloric acid, and more to learn about that in just a minute. So why is that relevant? How much of this gastric juice are we producing? 2 to 3 liters. 2 to 3 liters. So if we have 1.5 liters of saliva, we have now 3 liters of gastric juice per day. We now are accounting for almost 5 liters in one day worth of fluid. So in this process, I mentioned that the gastric juices contain quite a bit of hydrogen ions. Those hydrogen ions can come in and irritate the columnar cells. Here you can see those surface, the simple columnar epithelial cells. They can come in and start attacking those cells. So to counter that, those surface mucus cells, as well as the mucus neck cells, produce a bunch of mucin. And that mucin, once mixed with the water, creates this very thick layer of mucus. And that mucus separates the pH of 2 in the lumen with that of the surrounding tissues, which should be neutral at pH of 7. So this is fairly important when we look at how we maintain the external environment, in the acidic environment in the stomach, separate from that of the tissues. Because what would happen if we don't get the production of mucus? Well, those hydrogen ions can come in and start irritating those areas. And once they start irritating those areas, they can start attacking the cells. They can start attacking the submucosal layers, which are heavily rich in nerve fibers. That absence of mucus or that irritation of the submucosal layers are exactly what ulcers are. Ulcers are the breakdown of the epithelial tissue, exposing the submucosal layers and irritation of those areas. So looking at that, we mentioned that there's an acidic environment of the stomach, a pH of 2. Why is this important? Well, anything that you ever end up swallowing, if you think about any lower respiratory diseases of any kind, you've got pneumonia or anything like that, you're coughing up stuff. What do you typically do when you cough it up? If you're not spitting out, you're swallowing it. And if you end up swallowing it, why do we end up swallowing it? You're still inside your human body, but that acidic environment comes in and starts destroying those bacteria. The second thing we also see is the denaturing of the proteins. So those proteins that are present in the food start to unravel and they start to separate due to those hydrogen ions. Inactivation of salivary amylase. We're stopping amylase. We're stopping the breakdown of starches due to salivary amylase. We're activating lingual lipase, and we're converting pepsinogen into pepsin. So as I mentioned earlier, we have our gastric pits. We've got our mucus layer. We have pepsinogen that's produced, again, our pepsinogen produced by our chief cells. Our chief cells are going to come in, so here's our chief cell. And that's producing our pepsinogen. Pepsinogen is going to make its way through out into the lumen of the stomach, where the pH of 2 will trim off that excess amino acids and convert that pepsinogen into pepsin. And pepsin is now an active protease. And due to the presence of that mucus, that pepsin cannot come back and break apart the proteins of those columnar cells. It's a way of keeping it separate. The other enzymes, which you mentioned, gastric lipase, intrinsic factor, IF. Intrinsic factor is needed for the absorption of vitamin B12. Vitamin B12 is important for red blood cell production. The other thing I want to also point out, the hormones, gastrin, which we mentioned for the purposes of producing gastric acid, cholecystokinin, another hormone that's also produced that will signal the production of bile by the pancreas, as well as the storage by the gallbladder. And then secretin for bicarbonate, for the production of bicarbonate and the secretion of bicarbonate to buffer that. So once we make its way from the stomach to the small intestine, the buffering that takes place from the pH of 2 comes in with bicarbonate, and then we can neutralize the acids to bring us back to the normal environment. So if I were to take us back to that earlier slide where we look at what's taking place in, I don't know how far back I have to go, there we go. Once we make it from the stomach down to the small intestine, we want to be able to buffer that through secretin. And this is just going to show us the negative feedback loop that takes place. Is food entering the stomach. So food entering the stomach where we have a pH of 2. And there's two things that are happening from a mechanical standpoint as well as a physical standpoint. The physical standpoint is that the stomach expands. And there's stretch receptors that are receiving the stretch of the stomach. And that's all going to feed back into the submucosal plexus. So within the submucosal plexus, the ones that parasympathetic pathways innervate is going to now, so this is our control center, and our receptors. The afferent pathway are the stretch receptors that lead into the control center. Then the submucosal plexus will then send impulses that will result in the secretion of HCl. So the efferent pathway is a production of hydrochloric acid by their parietal cells. That'll also cause local contractions or mixing waves. To start churning through, okay? So play that back one more time. The presence of food in the stomach, which will expand the stomach, will then cause the production of acids as well as contractions taking place. So that's what's happening with the physical level, with the stretch receptors. The other thing that's taking place is through the chemical changes, namely the pH. Now you've got this stomach. I'm just going to draw up stomach. And here's our esophagus. We know that the pH is a pH of 2. And whatever we're swallowing down has about a pH of 6, 6.5. Now, whatever you just eat, once it makes its way into the stomach juices, what's happening to the pH as it starts to fill up? pH is going to rise. So if I have a normally acidic environment, and I start dumping, let's just say water, I start dumping, what I'm doing to that pH, or in other words, what I'm doing to that concentration of hydrogen ions, is I'm now diluting it. Right? I'm starting to fill up that stomach with water and food. Now the pH in the stomach is going to rise. And we have chemoreceptors that will detect the changes in the pH. Or in other words, detect the changes in the concentration of hydrogen ions with that of the entire volume. So the chemoreceptors detect the changes, and therefore again, lend that back to the control center, which will signal the production of hydrochloric acid. So the rise in pH, to sum that all up, the rise in pH detected by the chemoreceptors will trigger the submucosal plexus, which will then trigger it through the efferent pathway to produce hydrochloric acid by virtue of our parietal cells, to lower pH back down. So the rise in pH results in the lowering of the pH back to a pH of 2. What happens when we start producing enough hydrochloric acid? Once our pH runs right around 2, that's going to provide negative feedback to prevent the production of gastric juices, hydrochloric acid. So looking at that, at the stomach, as I mentioned earlier, the stomach is primarily digestion, and very little absorption actually taking place. But what type of absorption does take place? Water, ions, some short-chain fatty acids, and alcohol. If you ever, I'm sure after finals, I don't know, for those of you that drink alcohol, might take that first sip of alcohol, and you might notice that all of a sudden, maybe this is just me, but I feel it within 10-15 seconds. How does it make its way through? How do you absorb it so quickly? Well, not only are you absorbing in the mouth, there's a little bit of absorption taking place in the mouth due to the rich capillary beds there, but also absorption that's taking place within the stomach. Also, looking at water, and then combine that with some ions. What type of slurry have we seen that has water with ions in it? Gatorade is a good example. You have sodium chloride, which is salt. You have potassium in there. That actually increases the rate of water absorption. So that all takes place in the stomach. Other drugs that come into play, aspirin also is absorbed in the stomach. That's a place where you can absorb fairly quickly. Now, when you filled up that stomach, and again, thinking about what happens after a large meal, after you've eaten your meal, think about the rate at which you empty out your stomach. Anytime you eat just something sugary, let's say for breakfast, you had something healthy like a donut. Well, how's you going to use a donut? What's something sugary that you can eat for breakfast? Cereal?
Frosted Flakes.
Bread, Frosted Flakes, mainly high in sugar. Do you ever feel full off that? You might feel, if you ate a bunch of Frosted Flakes, you might feel full, but you might start feeling hungry again a little bit later. Why is that? Because carbohydrates don't spend that much time in the stomach. Now, take that meal and start adding some protein to it. And that might keep you fuller a little bit longer. Why? Because it takes a little bit longer to empty that protein out of the stomach. And now add something like fats. So take a very fatty meal, and it just sits, and you feel that feeling of sitting in your stomach for quite some time. So eat something like, it reminds me of I had a contest with my friends back in college of who could eat the most ribs from Tony Roma's, because they had it was like Thursday, like eat all you can eat ribs. You had like 4 big guys that came in, and you could see like the waiters are just like, oh no, it was disgusting. It's really disgusting. Anyway, the point is a lot of protein and a lot of fat after eating a gross amount of like 150 ribs or something like that.
Oh my God.
I never felt full I've never felt that full for that long before. I think it was like a full-on like day and a half I couldn't eat, and like still feeling full the next day. Why? Because it sits there in the stomach. It takes a long time. I mean, you're not only do you have a bunch of protein in there, a lot of fat, a lot of stomach motions that are the mixing waves that are taking place, and then also the the rate of gastric emptying that takes place due to the density of the food itself. So looking at the time, the gastric emptying that takes place, that's all governed by the pyloric sphincter, which is going to allow the quick squeezing of chyme into the small intestine. So in that process, we're going to start to navigate and move towards the process of digestion that takes place in the intestinal phase. So in this process, which we'll learn on Thursday, is everything that takes place with the small intestine. We're going to get pancreatic juice, we're going to get bile, and we're going to get all the other secreted enzymes that are going to dump into the small intestine. And this is how we continue digestion, again, especially within the first beginning, the duodenum, which is going to be the first 12 inches of the stomach. And if you look at the term duodenum, that's going to refer to, I believe that was 12 finger widths or something like that that you mentioned. It's actually the first 10 to 12 inches of the small intestine. So with that, you'll see the rate of stomach emptying, how we empty stuff out. And again, another thing we can look at is the rate of stomach filling. So with that, to kind of answer the question that we mentioned, that I heard a little bit earlier, is the different surgical interventions in reducing the capacity of the stomach itself. So we have different surgical interventions that have taken place, and some of them are more of the primitive ways, but the first one of it is called stomach stapling. Have you guys heard of stomach stapling? Yeah, this is actually one of the earlier ways of creating a very small pouch of the stomach. Now that you've learned the processes in which the stomach expands and signals the feeling of fullness, which we call satiety, the theory behind these surgical interventions is that if we reduce the size of the stomach pouch, you still get the stretch signals that take place that should still give you the feeling of fullness. But at the same time, if we could also limit the size, the capacity to distend or to expand, that also should give us the feeling of fullness. So what they've done in what's called vertical banded gastroplastic surgery is to staple down everything from the fundus all the way down to the mid-body of the stomach. Okay? So they create a hole, and they staple all around, and they staple up to this area, and then they create a band. They use a band around this area to limit the rate of food entering the pylorus as well as the lower body of the stomach. So as this distends a little bit, this is going to reduce the capacity of which they can consume, and then this is going to undergo the same process of gastric emptying. The next process is what's called Roux-en-Y type gastric bypass. Now, if you haven't heard of stomach stapling, have you guys ever heard of gastric bypass? Gastric bypass is actually the most invasive in all of them. And it's called Roux-en-Y. And what does Roux mean? R-O-U-X? It's a false limb. And basically what we're doing is we're completely excising the cardiac region of the stomach. And they're going to literally cut that entire thing off. They're going to separate the stomach from the esophagus. And what they'll do is they'll take now the jejunum, just downstream of the duodenum. They're going to take it, they're going to cut this area off, and they're going to attach that to this fake pouch that they've created. Hence the term fake limb. They're taking the jejunum and attaching it back to the stomach. So why do they do this? Again, smaller pouch. But then the question is, why don't they remove the rest of the stomach? Well, you can't really do that because you got a lot of arteries and veins and everything else. So you want to keep that intact. So all of this is bypassed, hence gastric bypass. You're actually bypassing the entire stomach. And you can't really cut off the duodenum because you've got all these other pancreatic pathways, which we'll learn later on, pancreatic pathways that dump into the duodenum. So anything that makes its way in here will make its way and dump right back into the jejunum. So they create, they'll section this entire area off, they break this part off, and they attach it to the stomach. And then they create a little anastomosis, a little portal that allows those pancreatic juices to dump back into the jejunum. So in effect, what this does, it limits the size of the packet and also spends less time in digestion within the duodenum. The last one, which I'll talk about, the last is called the last of the surgical intervention. And I got a video that I'll top off to show you the gastric bypass. Is the lap band. Have you guys heard of this one? Yeah, lap band. It's probably all over the news. You probably say it's been around for quite some time. The lap band is a less invasive surgery in which they can use laparoscopic surgery. It's basically tubes that go in. They can create very small incisions to go into the stomach and install a port right underneath the skin, which attaches to the gastric band, an inflatable cuff that wraps around the cardiac region. And that port, so that band, again, limits the rate at which the stomach fills. So this fake pouch that's created is the area that will store the food, and it'll eventually squeeze through and make its way into the rest of the stomach. So the theory behind this is not to cut anything apart, but it's to slow the rate at which we can fill the stomach, to give you the feeling again of satiety. They want to see if the typical things like diet and exercise don't work, and they would, before ever giving you the lap band, well, you'll have to have evidence that the typical conventions haven't worked to reduce your body mass index, which I won't really get into, but it's basically reducing your weight. Now the problems with the lap band is that obviously you've got a port that's installed right underneath the skin. It's not a permanent solution. People can typically report pain on the area where the band is at. You can also cause the esophagus to dilate. So typically people that have the lap band, now you've got all this excess pressure in the stomach pouch, which is going to exert more pressure on the lower esophageal sphincter. So typically people that do get the lap band can report higher rates of heartburn. So it's typically an adverse event that's expected. Another thing too is you can also get slippage of the gastric band. If that gastric band slips right back onto the esophagus, now your stomach is back to normal where it's at, and now you've got this band right around the sphincter. It can cause a lot of uncomfortability at that point. So with that, I'm going to leave you with the videos. If you are a little iffy at watching these videos, but if you are, feel free to leave, or if you just want to leave. But if you really want to just also watch how this takes place, it's a 9-minute video. I'm just going to cut to the juicy parts.
Call that jejunoplasty.
And
Is this reversible?
Yeah.
gastric bypass?
Wait, the lap band? or
Lap
the-
band,
oh,
yes.
lap band. No.
Gastric bypass, no. You'll see what they're doing right here. They're right there at the cardiac region, and they literally use this device. This device actually is really interesting because it's basically a heated wand that opens up like scissors, and then it clamps down, and it creates the scissoring motion where it will sew at the same time. So not only does it cauterize, but it also creates the stitches. So whoever created this particular device, incredible. Like a millionaire by now. So you see the excisions that take place. So now they've excised this whole area off.
Next, we're going to go down below.
Here you can see the the layers of fat. They actually call now the mesentery. It's actually now considered an organ. They just named it an organ. Here they're going through and measuring the small intestine. They're going to make a cut the jejunum from the duodenum.
And then we make another division in what's called the mesentery. The mesentery is this yellow fatty tissue that we're cutting.
They used to think that mesentery was just fat, but now they called it an endocrine tissue, or an endocrine organ. There it is.
Sometimes one of those blood vessels can bleed.
So they're cauterizing.
Now we're going to start to run the small intestine so that we can find
That's plumbing. They're glorified plumbers. Like orthopedics, glorified carpenters.
And we'll start construction.
That's a mouthful. Jejunum.
First step is to make a hole.
It's basically sticking two pieces together. They create a hole, stick them together,
Second hole in the other part of the small intestine.
and
This time we're going to put our
so this is the anastomosis, the hole that they're creating between the two.
so that we can
The jejunum attached to the jejunum, just downstream from the duodenum.
recall that the last time we stapled on the outside of the intestine to a split valve. Now we're going to staple inside.
Oh, they really put it inside.
And then they're gonna seal this all the way around. And watch as the device works. They've now sealed it together,
Now we still have a resultant hole here.
and now they're gonna take that and they're gonna clamp
So
it.
we're going to take our stapler one more time and close that hole. And now we have completed our jejunostomy.
So now they've connected the two.
Lift that file, and you'll notice that there is a defect here behind the file.
So when I get to the later part, what do plumbers do when they want to detect leaks?
See that hole right there?
Fill it up with water, which is exactly what they do. So they'll fill this whole area up with water. This is just kind of fixing a defect here. They're going to fill this whole area up with water, and they're going to push a bunch of air through to see if there's any leaks. Are they doing?
Pouch.
Oh no, they're still creating the pouch.
Pouch.
Here's the pouch that they're creating.
This extra tissue is hanging on by a thread, so we'll just use one more fire with the stapler to remove it.
See how the stapler works? Zip it up.
And now our gastrojejunostomy is completed.
So that's it. That's a gastric bypass. They created the pouch. Everything's stuck together now. They're going to keep the stomach in place.
There's just one more of those mesenteric defects right here.
And
Air bubbles.
so look, they filled it up with water,
Air bubbles would indicate.
and they're filling up. They're basically blasting air down into the stomach, and just checking for any leaks.
Air bubbles. So we know that the connection is airtight.
That light that you're seeing is a laparoscope that's on the inside. That's all it is, plumbing.
Now we simply suck out all of our water. And
All right, thanks for sticking through.
through,
And I will see you guys on Tuesday for a short week.
and our operation is complete.
Just taking a couple steps ahead. But the real thing I wanted to show you guys is just a summary of what we talked about. I'm going to come back to this slide. What I want to illustrate here, this slide is right after the parietal cell stimulation, where we technically actually left off. We left off last Thursday talking about the surgical interventions for such as gastric bypass, lap band, and the root and why gastric bypass. Now, I'm going to continue on to more the physiology of how we're going to produce hydrochloric acid in our parietal cells. As we talked about the two different types of cells in the stomach, we saw chief cells that produced pepsinogen that became activated in the low pH lumen of the stomach. And that pepsinogen was turned into pepsin, which were our proteases. Our proteases were the ones that would end up digesting proteins into smaller peptides or even down to their amino acids. So that's one aspect of it where we saw the enzyme. Now we need to see how we create the acidic environment of the stomach through the carbonic anhydrase. We'll take a look at that in just a little bit, but what I wanted to do just is to show you, just to do a quick recap of what we talked so far. So in our schematic, you can see we do have the entire alimentary canal, again, consistent with the external environment, in which anything that is ingested by the mouth makes its way through, takes its way through the cephalic phase. The cephalic phase, again, referring to the head. So here we are at the cephalic phase, which starts the whole process of the mechanical breakdown, where we use the mouth to break our food down into smaller pieces. We mash saliva, which contains the digestive enzymes, namely our salivary amylase as well as our lingual lipase. That mashes it right into our food, and now we call it the bolus of food, which starts the process of making its way into the involuntary phase of through the esophagus, into the stomach pouch, where the food is now introduced into the acidic environment. We have our pepsin secreted by our chief cells. We have our parietal cells contributing to the pH of 2 in what's called our gastric phase. And in the gastric phase, what we learned last Thursday is that salivary amylase is inactivated due to the acidic environment, but we see lingual lipase being activated in the acidic environment. What we're going to be covering today is everything that's taking place in the intestinal phase, of which we'll talk about the small intestine. Everything from the stomach, from the pylorus to the stomach, we'll make our way through the small intestine. You'll see we're making our way through the large intestine, and then we're going to talk about defecation. And we need to also talk about 3 accessory organs, namely our pancreas, our liver, and then right behind, tucked behind the liver, is our gallbladder. So these are considered accessory organs because, as I mentioned, food doesn't travel through these areas. So anything where food travels through is considered the alimentary canal. These are going to have exocrine functions in that they're going to produce things that will assist in digestion, specifically within the intestinal phase. Okay. And once we make our way back into the small intestine, we're back at a pH of about 6 to 7. Cephalic phase was around pH of 7. By now we have our enzymes, which we discussed, salivary amylase. We have lingual lipase. We have our pepsin. And then we'll talk about the other enzymes that are produced by the pancreas, the production of bile by the liver. We'll talk about a couple more liver functions as well, since we're already on that topic. And then the rest of the intestines we'll cover. With that, now we're going to look at, take a little step back and look at our cells, of which we discussed parietal cells, the cells that are going to produce hydrochloric acid. So what I want to talk about is the mechanism at which we produce HCl, hydrochloric acid. And the main enzyme that's involved in this whole process is the enzyme that we've talked about quite a bit, carbonic anhydrase. So our parietal cells contain lots of this enzyme. Because what's going to happen is, here you can see the lumen of the stomach. So this is our acidic environment. We're going to have that thick layer of mucus that's going to separate the acidic environment with that of the epithelial cells. And then outside of the epithelial cells, we'll have our interstitial fluid, and then we'll have the capillary that runs through. So everything from this point is considered extracellular fluid. we know that these epithelial cells, as well as the parietal cells, are not damaged by way of that thick layer of mucus that separates the high acidity from our normal pH for these cells to operate. So that carbonic anhydrase, again, our parietal cells that are embedded with these epithelial cells are going to convert through the process of CO2, carbon dioxide, plus water will convert into carbonic acid. And that carbonic acid will dissociate into our proton as well as our bicarbonate. So that process with carbonic anhydrase is the same. So what we need to talk about is how do we get these components, how do we get CO2 and water into the cell and through our conversion, how do we produce hydrochloric acid on the apical side of the parietal cell? So let's start off with our parietal cell. Parietal cell is metabolically active, and I'll show you where that step is taking place. And through that metabolically active parietal cell, we're producing carbon dioxide. We use ATP in order to produce carbon dioxide. Carbon dioxide mixes with water through carbonic anhydrase within our parietal cell will then produce carbonic acid. That carbonic acid will dissociate. Our hydrogen ion will make its way through, and then over on the apical side, we're going to see what's called a proton pump, similar to what you guys learned about for our sodium-potassium pump. But the key difference is we're allowing potassium in, but we're shooting out protons. And given that our hydrogen ions are moving from a low concentration of hydrogen ions, okay, so here the concentration is low, into a very acidic environment where we have a very high concentration of hydrogen ions. So we're moving against the gradient. We've got to pump those hydrogen ions out into the lumen. So therefore we're going to use ATP. This is active transport. And since we're pushing out a proton, we're also going to be pulling in a potassium ion, which actually will make its way right back out through these open channels. So potassium kind of circulates all the way through. Whenever you're pushing a positive ion up, you're going to pull a positive ion in, and you're going to keep the cell electrically neutral. But nonetheless, the main idea is to push protons out into the high concentration of hydrogen ions. So we've pushed that hydrogen out. Now, where do we get that bicarbonate? That bicarbonate that split off the carbonic acid will then shift out into the cell. Will diffuse from a high concentration over to a low concentration. And given that we're moving down the diffusion gradient, we're not using ATP. So here we're going to end up using what's called an antiporter. An antiporter pushes that negative ion out and pulls chloride in from the extracellular environment. So again, from our early lecture in cellular physiology, we mentioned that chloride ions are high in concentration on the outside of the cell. So from the interstitial fluid, it'll get pulled in. While we push bicarbonate out, chloride gets pulled in. And chloride again will move down its diffusion gradient down back into the lumen. So I'll touch back on that in just a little bit. Bicarbonate makes its way into the interstitial fluid, into the capillaries, to contribute again to the buffering capacity of our blood, to maintain a pH between 7.35 to 7.45. So that bicarbonate helps buffer any changes in the concentration of hydrogen ions. So if there's metabolic acidosis taking place or any alkalosis, we can prevent any changes to maintain that homeostatic pH in the blood. Now, what we left off with on the apical side of the parietal cell, we had hydrogen ion making its way out, and we had chloride making its way out. So get these two to mix together, you got a positive ion and a negative ion. These opposites will attract, and you get hydrochloric acid. So all the while, that parietal cell is continually churning out hydrogen ions and chloride ions. We're going to contribute to the acidic environment that's taking place in the stomach, and that's how we get a pH of 2. What questions do we have about this?
Where is this taking place?
This is taking place in the stomach. So this is the parietal cell embedded within the mucosa. So just taking a quick step back. Here you see the gastric pits. This is the epithelial layer of the stomach. We have these gastric pits, and deep within the gastric pits, we have our chief cells that produce our pepsinogen, and we have our parietal cells that produce hydrochloric acid. And it's going to make its way all the way up, and it's going to get diffused out into the lumen. Here's our lumen of the stomach, and here is that heavy mucus layer. All right, so now we know how the parietal cell turns on, or at least we know how it produces hydrochloric acid. Let's talk about how it gets activated. And as we mentioned, bless you, as we mentioned, digestion is a parasympathetic activity. Again, parasympathetic pathways, rest and digest. So the postganglionic neurons that innervate the stomach are going to be secreting acetylcholine. Not epinephrine. Epinephrine is used for the sympathetic pathways. Acetylcholine is going to be our main neurotransmitter. So we have cholinergic neurons that are present on the basal side of the parietal cell. And as the neurotransmitters of acetylcholine make its way through, it'll come in and activate and turn on our parietal cells. Parietal cells will then undergo the process of creating hydrochloric acid, and that contributes to the low pH of the stomach. So this should make sense. If the sympathetic pathways are activated, you're going to get a bunch of epinephrine that's going to make its way into the stomach, and that won't turn on the production of hydrochloric acid. It's only until we get the parasympathetic pathways that get activated that secrete acetylcholine, bind to the cholinergic neurons, that turn on the process of production of hydrochloric acid. So this should make sense in terms of the digestion ramping up through these pathways, as we illustrated before. That's one mechanism. The other mechanisms involve gastrin, and gastrin were secreted by the G cells we mentioned in our stomach slide. We do have G cells that are located that will produce our gastrin. Gastrin will come in and work locally. It's not working through the local bloodstream. It'll work locally to come in and activate those parietal cells. Those parietal cells become activated and therefore again produce hydrochloric acid. The last one of these is histamine. And histamine is a paracrine hormone. And these are actually produced by cells much deeper within. We do have other layers contained within, namely the lamina propria. So if you read through the book, it'll mention histamine. And the lamina propria will then, there's specific cells called mast cells that will secrete this histamine. And histamine, again, a localized response that would also activate the parietal cells to produce hydrochloric acid. So all of these are a local response. So that's really it that we need to talk about in terms of the two cells, namely your chief cells as well as your parietal cells, and then our G cells for the production of our gastrin hormone. The other hormones that we want to talk about have major control over appetite. So leptin and ghrelin. Leptin, the first one we're going to talk about, is specifically a hormone that's produced by fat cells. Leptin produced by fat cells, ghrelin produced by stomach cells. And they work by giving us sensory information that's led into that will tell us whether we need to increase our appetite or decrease our appetite. And leptin is something that, like I said, is produced by the fat cells for the sole purpose of decreasing your appetite. So this should make sense in a sense where in obese people typically have more fat cells. So fat cells will produce excess leptin, and that excess leptin should then decrease appetite. So those that are typically thinner, that have less fat cells, will typically have a higher appetite. Okay? And from an evolutionary perspective, that should make sense. If you don't have enough body fat, you're going to have less leptin. And the less leptin, the less capacity you have to decrease your appetite. So in other words, thinner people are hungry. Okay? So in that sense, what that whole purpose of leptin is to let you know, pretty much to govern your level of your appetite. Now, what I want to talk about is just a story back in the 90s when they discovered leptin and ghrelin. They had experiments that they worked on what are called knockout mice. And they take the gene of their standard lab mice, and they found the gene that encodes for leptin. And what they noticed about these particular mice that didn't make leptin, they were severely obese mice. Okay? And that should make sense, because if they can't produce leptin, they constantly have an appetite. And these mice would just consistently overeat. But what they did was they gave leptin back to these knockout mice. They inject leptin back in there, and they found that these mice all of a sudden started losing weight. They were able to regulate their appetite, and then over the course of a couple weeks, they were back to their normal weights. So Amgen caught wind of this whole study that said leptin has the properties to reduce body fat. And so Amgen bought the rights to leptin. They created their own version of leptin and basically dumped in, I think that if this was back in the 90s, they dumped in about $20 million into this whole project. And this leptin would be theirs because they thought they found the secret for bringing weight loss, essentially, bringing down body fat in people. So they, as you know, we go from mouse trials and then we start to bring it out to clinical trials. We had clinical trials as a result. Amgen started their clinical trials on obese people, gave them leptin, and found out technically it really didn't do anything. So this was kind of a $20 million sunk into something. But what they did find was that it wasn't necessarily leptin. Remember, leptin is a hormone, but in order for that hormone to do anything, it needs to bind to a receptor. So in the cases where we see obesity, where it's not the leptin that's an issue that they discovered with our knockout mice, leptin needs to bind to a receptor. And the issue where in cases of morbid obesity, why their appetites were not regulated, even though they had normal and even actually technically excessive levels of leptin, they found that the receptors were constantly bombarded by this leptin. And the receptors no longer could receive leptin. Again, kind of tying into the whole thing of leptin receptor resistance is a term I'm looking for. Very similar to that of insulin resistance, like type 2 diabetes. Type 2 diabetes, you can produce insulin just fine. In fact, you're producing a lot of insulin. It's just the receptors aren't binding as optimally to the receptor. So in order for this leptin to take place, it needs to bind to the receptor. If the receptor is constantly bombarded, it loses its ability to bind to leptin. So the issue that was skirted by the people that studied those knockout mice was that it had nothing to do with leptin itself. It was all due to the leptin receptor. So in these cases, this leptin receptor resistance is basically saying, in the cases of morbid obesity, their fat cells were constantly producing leptin, but they couldn't bind to the receptors to decrease their appetite. Okay? So this kind of ties into the whole thing of, and looking at normal weights, the effect of leptin is to tell you you're full, okay? And that will regulate your appetite at that point. Now, consistently, if you override this process, you'll start producing more and more leptin to try and shut off this process, to to decrease your appetite, and therefore can come in and start overbinding to these receptors. These receptors create the receptor hormone complex that needs to get degraded on the cell membrane, and therefore this process isn't optimal for decreasing appetite. So that's the process of leptin. But the other side of it all, we need to talk about what's going to stimulate the appetite, and that's where ghrelin comes in. So ghrelin is produced by the stomach cells. It receives information from stretch receptors, things within the stomach. It's receiving information from the central nervous system, letting us know that the process of appetite stimulation is ramping up. So with that, when we measure ghrelin levels, let's say throughout the day, where would we typically see the highest levels of ghrelin? You would expect high levels of ghrelin right before feeding. Okay? And in fact, they've done studies on those that do intermittent fasting. And intermittent fasting, I know I may have just talked about prior, but intermittent fasting is a period of about 15 to 20 hours of fasting. And they found that ghrelin levels are very high right before feeding. And that should make sense. Ghrelin levels are high, you've got a very strong stimulant letting you know, bring up your appetite, you're about to eat. Now, as we start to fill up our stomach, so the chemoreceptors are now picking up the changes in the stomach, the stomach is starting to fill up, and the stretch receptors are now coming in and also feeding the central nervous system, letting us know that our stomach is filling up. And that will do the exact opposite, where it'll come in and inhibit the process or the production of ghrelin to reduce your appetite at that point. So looking at these two, leptin and ghrelin, basically are antagonistic hormones. They work against each other. One will decrease the appetite, while the other will stimulate the appetite. And I don't believe the book goes too much into this. This is something that I wanted to kind of throw in, because these are key hormones that were discovered, I wouldn't say recently, but within the 90s and 2000s. The next slide really just goes over looking at the two, the ghrelin, the appetite stimulator, and leptin, the appetite suppressor. And they work hand in hand. Leptin, which is going to decrease your appetite, again, taking place after you eat. Ghrelin will be the highest right before you eat. And they work hand in hand in order to stimulate your need to feed. All right, we've talked about pepsin so far. The other one I forgot and neglected to mention is gastric lipase. And we'll continue on with the rest of the intestinal phase. And first, what I'd like to talk about is our pancreas. So we've talked about our liver and our pancreas as our accessory organs. Let's take a look at the pancreas. Now the pancreas, which you can see, is embedded right below the liver, nestled up right against the duodenum of the small intestine, which we'll talk about when we cover the small intestine. And the key thing about the pancreas is that it produces our enzymes. We've got a couple pancreatic enzymes to talk about, and then the presence of high amounts of sodium bicarbonate. And that aims to buffer the chyme that makes its way from the pylorus of the stomach. Here's our pyloric sphincter. And we're going to dump all this acidic chyme into the small intestine. So the pancreas is producing that buffer that comes in, that bicarbonate that comes in to neutralize the acidity of that chyme, to introduce us back to a neutral pH. So here you can see the production of those, again, our enzymes and our bicarbonate, and we're going to continue on with those enzymes. We're going to continue on with digestion in the small intestine. More to come on that. The other portion that we want to talk about the pancreas is that the output, we'll look at in the next couple slides, we'll look at the functional units within the pancreas for the exocrine functions. And we're going to see how those enzymes, as well as our sodium bicarbonate, are going to make their way out and to the primary output. And that primary output is called the hepatopancreatic ampulla, or the ampulla of Vater. And why is it called the hepatopancreatic? Hepato meaning liver. Here you'll see the liver for its pathways for producing bile. It shares the same output as that of the pancreas. And you get this little nipple right there that's embedded within the mucosa. And the purpose of that is to basically dump out the contents of the enzymes, as well as bile produced by the liver, as well as the sodium bicarbonate produced by the pancreas. You also do have other outputs called the accessory duct, so the duct of Santorini, but the main output would be that hepatopancreatic ampulla. And where this takes place, it's about 4 inches. Here you'll see the length of the duodenum is about 10 inches. The first 4 inches are going to be the point at which we're going to dump these. To kind of tie in back to what we talked about last Thursday, when we saw the root and wide gastric bypass. There was that question that I sort of asked of why, with the presence of the stomach, you saw the esophagus into the stomach, and from the stomach pouch, the remnants of that stomach was still attached to the duodenum. And you saw that we had an anastomosis that attached that little pouch, that little stomach pouch, and connected all the way to the jejunum. What was the sole purpose of this duodenum if we're completely bypassing it? The whole purpose of that is that the pancreas and the liver still need to do their job. And again, to produce the bile, the enzymes, as well as the sodium bicarbonate that are going to get dumped into this region and make its way down to the jejunum where we had that other attachment. So we don't want to get rid of this, because if we sever that duodenum, we have no way of using the enzymes that are produced by the pancreas as well as the bile for that continued digestion. So any way you look at it, this is where that action takes place. So let's take a step back and look at how we produce the stuff that the pancreas produces. We're going to backtrack to what we looked at before when we looked at our acini. So the pancreatic acini are going to be the exocrine cells. We're going to zoom in for the pancreas. We're going to zoom in into this little cluster of grapes at the very ends of each of these pancreatic ducts. And within these, we'll zoom in even further, and you can see our cuboidal cells. You can see all of our endocrine cells, of which we have the alpha cell, the beta cell, and our delta cells, all embedded. So this is our endocrine. And the acini on the outside, in yellow, are going to be our exocrine cells. And it's these cells that we want to focus on now, because these are the ones that are going to be producing the pancreatic juices, which are going to contain the enzymes as well as the sodium bicarbonate. So these cuboidal cells are tasked to produce those. They do their production. Here you can even zoom in a little bit further. You can see the production that will dump into the ducts and therefore make their way over to the hepatopancreatic ampulla. Now, looking at the other endocrine cells, just as a recap, you can see our alpha cells, which are going to produce our glucagon, our beta cells, which are going to produce the insulin, and the delta cells that are going to produce our somatostatin. And where did we see somatostatin? We saw that in our endocrine lecture. Somatostatin was our growth hormone inhibiting hormone. So the purpose of this growth hormone inhibiting hormone, remember our growth hormone was responsible for the breakdown of carbohydrates and fats while preserving proteins. So in the case of somatostatin produced by the delta cells will come in and turn off the production of growth hormone. And that comes back over to our hypothalamus, to the pituitary, to prevent the production of those. So that's where we get the inhibition of growth hormone production. Looking at the makeup of the pancreatic juice, as I mentioned, enzymes plus sodium bicarbonate. How much of it do we produce a day? Is about 1.5 liters of alkaline pancreatic juice secreted right into the duodenum by way of the hepatopancreatic ampulla. So just to sum up the total volume of our juices, We had 1.5 liters a day for saliva. We had 3 liters for that of gastric juices. Now we have another 1.5 liters in pancreatic juices. So by now, we have 6 liters total of fluid that's dumped into the alimentary canal. And obviously we're going to have to reclaim that water. And where's that going to take place? That'll take place in the small intestine, but I'll talk about that when we get to that section. So the whole purpose of the pancreatic juice is to, again, buffer that pH. We have the acidic chyme that's making its way into the small intestine. We want to be able to buffer it, and that's where we get our sodium bicarbonate. Or rather, that's the whole purpose of the sodium bicarbonate. The other enzymes, you'll see, we have pancreatic amylase. Again, amylase for the purposes of digesting starches. We have our polypeptide enzymes. So if this is our starch, or these are our carbohydrates, our peptides, our polypeptide enzymes for proteins, and the last, pancreatic lipase for the fats. Looking at these, you'll see 5 polypeptide enzymes. I know you only see 4 on there, but the 5th one, which I'm not really ever ask you, is called enterokinase, or also known as enteropeptidase. So there's actually 5 enzymes: trypsin, chymotrypsin, carboxypeptidase, elastase, and then lastly enterokinase. So the acronym here that they use in medical school is ETCCE for eat, tender, chicken, chunks, elegantly. Now, these are silly, but that's how you remember these things. So these ETCCE is what's used, and that helps you remember elastase, trypsin, chymotrypsin, carboxypeptidase, as well as enterokinase. And I won't really go into the real main differences of all these. The main 4 right here, the whole purpose of this is really just to digest your proteins. You have long proteins from the food you've just consumed, and we want to be able to break that down through catabolic reactions into smaller peptides. Those smaller peptides, once we break them down into the smaller peptides, then down as that food travels down the small intestine, we're able to take those small peptides, absorb them through into our bloodstream, and then construct bigger proteins from that. So we want to create small peptides as well as the amino acids that come from it. So these help cleave these long proteins. They look specifically for a primary sequence of amino acids, and they'll just cut. That's all they do. And they continually cut until you have smaller fragments. Now, technically what's happening is we actually have that enterokinase or enteropeptidase. One of the things that I won't really go too much into is that trypsin is not actually released as trypsin. It's actually released as trypsinogen. Just like you saw in the stomach for pepsinogen, just like you saw angiotensinogen in our endocrine system. Enterokinase comes in and finds trypsinogen and cleaves off a bunch of proteins, leaving you with a very small enzyme called trypsin. So this is where these come in to cleave and turn it into trypsin. And trypsin is now activated to help cleave your proteins. That's really it with the pancreas. The pancreas doesn't really do anything else for the purposes of digestion other than produce all of these enzymes. The other enzyme that we mentioned is pancreatic lipase. And again, looking at the term lipase, breakdown of fats. So we have now lingual lipase activated in the stomach along with gastric lipase, and now we have pancreatic lipase to help create fatty acids and break apart those ester linkages. Now the liver is the other organ that's going to be involved in the production of bile. But since we're already here, we'll also talk about the other functions of the liver for the breakdown of red blood cells. You'll see that it's the largest gland, but it's the second largest organ. Do you guys know the first largest organ? The skin, yeah. So liver is right there, right as the second largest organ. We have two main lobes, which we won't really get into. But if you're going to take a look at the input and output of the blood, for the purposes of the liver's filtration of the blood, we've got to take a look at the input as well as the output. The first of the input come in from the portal vein. And this portal vein is a light purple. And it's a light purple because it is actually a bit of a mix of oxygenated blood with deoxygenated blood. And it's coming in from the veins and capillaries that surround the small intestine. So this portal vein that's coming in is going to have nutrient-rich blood, but deoxygenated blood. Now the hepatic artery, which comes off the descending aorta, the hepatic artery will feed in nutrient-poor blood, or nutrient-poor but oxygenated blood. So 75% of the blood that feeds into the liver comes in from the portal vein, the remaining 25% from the hepatic artery. So these feed into, and you can see the output, they basically will spiral together and feed right into the functional units. So we'll take a look at the functional units. The output of the blood that comes in from all of our input, once it bypasses our functional units, will then converge into the hepatic vein. You've got the central vein system that will all verge together into the hepatic vein. Hepatic vein will then dump into the inferior vena cava, returning us back to the right atrium, making its way into the pulmonary circuit. So knowing that we now have two inputs for the blood, we can see where that blood makes its way. So as I mentioned, portal vein and hepatic artery branch off into a bunch of smaller arteries, arterioles, into capillary beds. And within those capillary beds, there's going to be embedded within our functional unit called liver lobules. These liver lobules are a bunch of repeating hexagonal units, and you can see that on our image on the top right. A bunch of hexagons embedded, where right in the very center, you're going to see what's called the central vein. And the central vein is going to be something where it's going to be the drainage into the output. So this represents our output of the heart. Output again being on its way to the hepatic vein. You get these central veins that are going to be collecting blood. Let me first kind of clarify what I'm talking about here. Our portal vein, you can see, is our input of the heart. Input of the liver, I mean. Input from the portal vein, we get 75% of the blood through the portal vein. We get 25% of the blood through the portal arteriole. And they come together and are going to be all present at each of the single points on that hexagon shape. So each of those 6 points in the hexagon are going to consist of what's called the triad. And the triad, if you read about the triad, will tell you about the portal venule, our portal arteriole, again, branches of our input of blood, and then the last will be our bile duct for the purposes of producing bile. So these three are typically together on each of the sides of that hexagon. And the flow of the blood flows in from the portal venule as well as the portal arteriole, will flow into these sinusoid capillaries, which I'll describe in just a bit, and they'll drain into the central vein, as you can see right in the very center. So you see this point at which you get a bunch of spirals that are making their way in. They're all converging onto that central vein that will jump in and join these interlobular veins, which will all dump into the hepatic vein back to the inferior vena cava. So you can really see how the liver lobules serve to act as a filter. Everything from this, our triad, over down to the central vein. So what's taking place right in between? Again, we're going to have blood that's flowing through, and then we have the production of bile, which we'll see in just a bit. Now let's zoom in, and here you can see our triad again, our hepatic artery. You'll see that this was our portal vein, and then our bile duct. So here you can see the venous blood that makes its way in. They're going to now converge into these sinusoids, and then as they make their way through the sinusoid capillaries, they'll drain back into the central vein. These liver cells, and these liver cells are called hepatocytes. Hepatocytes. Oh, it's right there labeled off to the right. Hepatocytes are going to be these liver cells that are going to have numerous versatile functions. So looking at this pathway of blood, you're making its way through. You've got these capillaries, and you've got all these hepatocytes that are going to come in and assist in the breakdown of stuff that's present within the blood. So there's this definite filtration as it makes its way through. And then once you've cleaned up your blood, you're going to drain back into the central vein to return that back over to the inferior vena cava. So this should make sense where anything that you've just consumed, anything that whatever makes its way into the small intestines, gets absorbed through, we want to be able to come in and remove things that shouldn't be present in the blood. All right, so let's talk about the hepatocytes. Each of these individual cells, these red dots right here, what purposes do they have in terms of filtration? We have two functions we need to talk about, digestion and then non-digestion. The digestive purpose is for the synthesis, transformation, and storage of protein, carbohydrates, and fats. Again, blood coming in from the portal vein, from the small intestine. Here the liver can come in and store things that we might have in excess. So if you've just taken in a bunch of glucose, that glucose is going to make its way into the portal vein. It's going to raise blood sugar. And what better way to store that excess glucose within the liver? And that storage process, we mentioned, we want to create liver glycogen. And how we produce glycogen is through the process of glycogenesis. This is also where we get the process of gluconeogenesis, where any of that lactic acid that makes its way through, the liver can convert any of those back into available glucose. And if that glucose will be returning back into the bloodstream, it'll make its way into the central vein. Otherwise, if we want to store that glucose, we can store it as glycogen within these hepatocytes. Not only can we store glycogen, we can store proteins, and we can also store fats as well. This is another area, aside from fat cells, adipocytes, that we can actually store fat in. Has anybody here ever had, I'm probably butchering the name of it, foie gras? Yeah. Fatty duck liver? It's kind of a fancy food that I've never personally eaten. I don't think I'd ever eat it, but typically what they would do is they would take geese and they would feed them very high-fat diets. And because they've had such a high-fat diet, they end up storing a lot of that fat in the liver. And they take that liver, they crush it up, create like a pate out of it, and then serve it. I'm too rich for my blood. I've never eaten it, but nonetheless, that's the whole purpose of it. Take something in the case of you have a fatty diet, and you get a fatty liver. Okay, so that's what we do with it. The other purposes of the liver is detoxification. What are we detoxing from? Drugs and alcohol. Anything that's taken in that makes its way into our circulatory system needs to get broken down. And that's exactly what the liver is supposed to do. Now, abuse that system, and your liver is constantly working overtime, and will start to break down over time, and you're going to see what's called something called cirrhosis that you'll be able to explore on a later slide. We can also modify some of the molecules that are taken in, and then we can excrete anything that we don't need. The non-digestive is the phagocytosis of old or worn out cells. What worn out cells are we talking about mainly? Our red blood cells. Our red blood cells, as you remember, have about 100 to 120 day life cycle. They don't have mitochondria, they don't have any nuclei, so they can't regenerate their own cells, they can't repair themselves. After about 100, 120 days, they live out their life cycle, and we're not going to just dump out red blood cells out of our body, we're going to want to break that down. So where you saw the sinusoid capillaries in the bone marrow, where we saw the production of red blood cells, you're going to see the same sinusoid capillaries present within the liver lobule for the purposes of taking that red blood cell, breaking it apart, and reusing that for the new production of red blood cells. Okay? So we're going to break those old red blood cells down. The other things that the liver does, also produces a lot of our major proteins. So the major proteins that are present in the blood plasma, things that are dissolved in the blood plasma: heparin, prothrombin, fibrinogen. These main three things: blood clotting. Albumin, you've seen albumin before. That was our lipid-soluble hormone carrier. And then lastly, of all of these, you've definitely seen angiotensinogen. Angiotensinogen was the protein that's present in our plasma that in the process of the RAS system, where we get either dehydration or low blood pressure, angiotensinogen is converted into angiotensin by way of renin. Renin produced by our kidneys. So it'll activate it and turn it into angiotensin, and angiotensin will go through and get converted into angiotensin 1 to angiotensin 2, angiotensin 2 to aldosterone for raising blood pressure back up. So these are our non-digestive proteins that the liver produces. The other main thing that we'll talk about on the slide later is the production of bile. And I'll talk about that on the later slide. Here you can see again the pathway of blood, where we saw 75% from the portal vein, 25% from the hepatic artery, makes its way into the liver. This makes its way to the triad, into the liver sinusoids. Sinusoids then drain back into the central vein. So here you see these sinusoids all within. These are the little passages in between. You see the hepatocytes embedded right around the sinusoid capillaries. The sinusoids then converge onto the central vein, where from the central vein they converge into the hepatic vein. Hepatic vein is the one large vein that dumps right into the inferior vena cava. And that makes its way back into the right atrium to continue on with the pulmonary circuit. The other thing I want to look at are the other cells besides the hepatocytes. We also have fixed macrophages called Kupffer cells. And Kupffer cells, for their purpose is just basically to filter out any red blood cells, white cells, and any bacteria within the blood. So macrophages help basically clean up that blood. The other function that we need to talk about is the production of bile. And these hepatocytes, and keep in mind, I did mention a lot of different hepatocytes, or I'm sorry, a lot of functions of the hepatocytes. The functions of the hepatocytes really differ based on what thing they're next to. So the hepatocytes that are right next to what are called the bile canaliculi are the ones that are going to be producing bile. And bile is another exocrine product that help emulsify fats. And I'll talk about that on the next slide over. The emulsification of fats is our way of basically mixing fats into water. As that bile gets dumped into the duodenum, this is our way of our absorption of fats, and that's where bile is absolutely necessary. Now, how much do we produce in terms of bile? Now we have 1 liter of bile per day. So our running total for the amount of water that we've dumped into the system is now 7 liters. 1.5 in saliva, 3 in gastric juices, 1.5 in pancreatic juices. Now 1 liter of bile. That's 7 liters now. Per day. Now, what exactly does bile do? Well, bile, like I said, emulsifies fats. And the way it works, it has both hydrophilic and hydrophobic regions. And it comes in and circles around a lipid molecule. So here's our lipid molecule. It's all clumped together because this is a rather large molecule. And bile salts come in and will take that lipid and break it apart. And that breaking apart takes these little lipid droplets and encapsulates them in bile. So what exactly is the process of emulsification? If I can help you understand what emulsification is. Emulsification is taking fats and being able to dissolve that in water. So if I took oil and I took water and I put them together, they'll separate. Obviously you'll get the fat on top, you'll get the water separately. And that's not really going to be useful. Imagine that making its way into the small intestine. We have no way of taking those lipids, which are valuable, we have no way of absorbing that huge chunk of lipid. All those fats are stuck together. So what we need to do is to cleave all those lipids that are all stuck together and make them into smaller droplets. Those smaller droplets allow for increased surface area so that we can individually absorb the bile as well as those droplets. And we want to introduce that back into the circulatory system for use later on so that we can store those fats. So what exactly is the process of emulsification? Let me give you the example of an egg. And if you crack open a raw egg, you'll see the egg white and you'll see the yolk. And those two are typically maintained separately. It's because the egg white is mostly albumin, it's water soluble, but the yolk is going to contain a bunch of cholesterol, it's going to contain a bunch of fats in there, and those two don't mix. How do you mix them together? Well, you get a whisk and you mix it really well. And then you notice now it's all kind of mixed together, but technically they're not really mixed, they're just kind of embedded within each other. If you left that solution to sit for a little while, you'll notice that they'll start to separate after a little while. Another thing is milk. If you ever look at milk, milk is a combination of water as well as proteins that are dissolved in that water, but there's also fats in there. So if you ever look at whole milk or 2%, maybe not skim milk, but if you look at milk, milk is an emulsion. You've got fats dissolved in water. Let that milk sit out for a while, and you see a clear separation of fats on top of the water. So in this process, we're doing exactly that. We want to take those droplets of fat and mix them into the watery aqueous environment that it's going to make its way into once we're in the small intestine so that we can absorb this stuff, because this is important. This is tons of calories in our lipid that we can use to make our way back into the digestive system. Without these bile salts, if you have less bile, and why would you have less bile? This would be in the case of anybody that's ever had their gallbladder removed. So that is, which we'll call later as a cholecystectomy, is the removal of the gallbladder, which is supposed to concentrate bile. And I'll talk about that concentration of bile in just a bit. So in those cases where people have had a gallbladder removed, surgically removed, they have less ability to absorb fats. So typically people that have had their gallbladder removed will be advised to modify their diet, to not take in as much fats. And why is that? Because you have no way of emulsifying the fats. They'll make their way into the digestive system. You've got a bunch of lipids that are stuck together without that excess of bile, and that could cause some gastrointestinal distress within the large intestine, all due to the bacteria present that cannot react to a bunch of lipids all stuck together. Now, the other thing about bile, the dark pigmentation that comes from bile, is called bilirubin, and that comes from the breakdown of these old red blood cells within those hepatocytes. So the hepatocytes over here can come in and you have these sinusoids that come through. We can take in those red blood cells and break them apart. And that breaking apart, that byproduct is called bilirubin, and it comes from breaking apart those iron particles within hemoglobin. So that bilirubin, which you'll learn later on, is what contributes to that brown color of feces. Most of that, where that color comes from, why is your feces brown? It's bilirubin. The thing of why you're seeing this image to the bottom right, has anybody ever seen this before? For Lay's Light? Still out on the shelves, you don't see too much of it, but this was something that was heavy around the 90s and early 2000s. It was a product where they were able to find a so-called fat substitute, but it wasn't so much a substitute. It was called olestra. And olestra was a molecule that came in that also had the ability to interfere with bile salts. And the theory behind it was, and I've been describing the whole process of taking lipids and breaking them apart into these smaller particles that can be absorbed. But what if, in the purposes of diet modification, what if you don't want to absorb those lipids? You get this molecule called olestra that interferes with the binding, and now these lipids don't get absorbed. And eventually it'll make its way through the alimentary canal, and you can eliminate a good amount of fats without actually absorbing it. So this company created olestra for that purpose, and they started to introduce it to foods as a way to reduce the amount of fat that you absorb. Now that you don't have this emulsification, what happens in a lot of these cases after they've released this product is that some of the things that they found about people that ate a lot of this stuff is that fats are extremely important, not because they contain calories, but because there's also fat-soluble vitamins, namely vitamin A, D, vitamin E. They're all fat-soluble, and they need fat in order to dissolve it so that you can get that into your system. Now, if you can't absorb that fat, you don't get those vitamins that you need. And the other thing that's also involved is that those lipids that make their way through the large intestine cause a bunch of gastrointestinal distress. And one of the common side effects of people that ate these particular olestra-containing products had what was a condition called steatorrhea. And it kind of looks like the term diarrhea.
Fat in the, it's pretty fat in your
Extreme, yeah.
There's a
It's,
certain fish that does that too, like whitefish,
uh-huh.
that if you eat too much of it, you have that.
Really? What
Yeah.
kind of fish is that?
It's whitefish. If it's
So-
too much whitefish, it's like too much fatty fish.
Oh,
My
okay.
dad had it, so I knew about it.
Interesting, all right. Well, thank you for sharing. That's basically what Nancy was describing is the easier term for this is called anal leakage. It's the inability to allow fats would end up leaking and separating from the feces and will end up causing what's called anal leakage. And
They mentioned it in a movie.
what's that?
They mentioned it in a movie.
Through olestra?
They mentioned the anal leakage from chips. He's like, don't you know those chips give you anal leakage? I'm like, what? It's on the warning label.
Yeah. So the warning label, which has like a fine print in the very back that you don't read.
That's so funny.
Yeah. So that was something common, and they found people that were staining their underwear. And I won't really go into too many of the details, but it was just something common. They didn't quite pull it on the shelves. I know I've seen them around. I remember seeing at the very bottom of the chips aisle and saying, damn, they still sell olestra. And it's there.
That happens with Ally too, the diet pill or whatever.
What was it called?
Ally,
Ally? How do you spell that?
ALLI.
Okay.
I had a coworker that used it, and she said when she went to the bathroom, you could see the fat.
Oh yeah, is that the dieting pill?
Yeah.
My friend did that. She told me about it. I was kind of weirded out. Like,
She lost like 100 pounds, which she
Wow.
gained back. But she was like, when every time she went to poop, anything, because you were supposed to have a high fat diet, she's like, it all came out.
Oh, gross.
Yeah. It's like,
So, yeah, avoid these products. For the sake of yourself and for the sake of your underwear.
is it here?
That's what's funny is I remember when I was taking physiology and I learned, I didn't learn about olestra until I started reading more and more and started putting these slides together, and I'm like, I remember eating these Illustra chips, and I remember, and I started reading more about it. It was one of those, oh, that's why that happened. I could never really explain it, but thank you,
It's like the
physiology.
sugar-free gummy bears.
Sugar-free gummy bears?
The Haribo sugar-free gummy bears give you some sort of like awful diarrhea too, like explosive diarrhea. If you go on Amazon, you can read the comments of people that just, explosive diarrhea.
Wow. And I think people take also Amazon pictures too. I've seen. Anyway, got it. So stay away. So, modification of your diet really should be taking place prior to introducing it into the alimentary canal, as opposed to interfering within the system. Because as we've kind of been able to illustrate, anything that comes in and changes the process, especially of that of fat absorption, can really cause some major changes that we don't want to happen. As I mentioned, so now we're on the production of bile. We know that the liver has all these other functions, but we want to focus on the digestive portion. The bile canaliculi, which are present right in between the hepatocytes, come in and start dumping that bile that's produced into these hepatic ducts. You've got the left hepatic duct and the right hepatic duct, which all converge together into what's called the common hepatic duct. And the common hepatic duct then binds to joins that of that called the cystic duct. So gallbladder, that's off to the bottom of the liver, right behind the liver, will come together. So cystic duct and the common hepatic duct come together as what's called the common bile duct. And common bile duct then merges with that of the pancreatic duct to create what's called the hepatopancreatic ampulla. So with that, we see the process of where bile is dumped into the duodenum. Let's talk about how, and here you can see how this come together, our common bile duct as well as our pancreatic duct. Here you can see the hepatopancreatic ampulla that comes in and merges right into the small intestine, penetrates right through that mucosa in order to dump the pancreatic juices, the sodium bicarbonate, as well as the bile right into the small intestine. What's also present is this sphincter. So you do have this muscle layer, the mucosal, the muscularis layer is present with this smooth muscle that wraps around that particular ampulla in order to regulate the release of bile. So in this process, we're not constantly releasing bile into the small intestine. We do have to regulate it. So in the cases where in between meals, where we don't have food passing through, that sphincter is typically closed up. And by closing it up, that's going to now close off or occlude that ampulla, which will cause backup of this bile all the way through. So that backup will make its way all the way back up. And once it backs up, you'll have where the gallbladder comes in. You'll get the bile that backs up all up into the gallbladder. So we've got this watery bile that makes its way into the gallbladder, and the gallbladder will help in concentrating that bile by removing the water as well as the ions that are present within that bile. So now we get this concentrated bile that backs up, and what happens as food travels down, food makes its way into the stomach. The stomach now is going to start feeding information back into the central nervous system. You're going to have hormones traveling through, and the hormone that's going to help release the gallbladder, cause the gallbladder to contract, is CCK, and I'll show you that in our next slide over. So here you can see the back of the liver. You can see the gallbladder. CCK, cholecystokinin, is what's going to cause the release. And I talked about this a little bit in our stomach slide, where CCK comes in to contract the gallbladder. So why is it called cholecystokinin? If you break down the word, cysto means gland, chole means bile, and kinin, like kinesiology, is to move. So bile gland move is basically the name of it. So this hormone comes in and causes that gallbladder to contract and to secrete that concentrated bile to make its way down the common bile duct to dump right into the hepatopancreatic ampulla. So in those cases where if you've ever had a gallbladder removed, is what's called a cholecystectomy. Again, looking at chole for bile, cysto for gland, tectomy, which is the removal of. And typically, as I mentioned earlier, typically a modified diet, a diet lower in fat due to the lack of concentrated bile within the gallbladder. Now, even though you have less bile, you still have other lipases involved. Again, lingual lipase, gastric lipase, as well as pancreatic lipase. And if you go back into your homework, you don't have to bring it out right now, but your homework will look into summating all of these enzymes that we've talked about so far. So that way, that serves as a good table for you to kind of help summarize your notes and then piece it together in preparation for the final, at least for the new content within the final. All we've talked about so far was our pancreas. We've talked about our liver as well as the gallbladder. So we're done with our accessory organs. We're going to continue on through the journey through the alimentary canal by finishing up the small intestine and then talking about what happens in the large intestine. And the small intestine, we namely divide the different areas of the small intestine by looking at the duodenum, the jejunum, as well as the ileum. So you heard these terms when we talked about our, specifically our gastric bypass. We saw where the jejunum comes in. But the duodenum is going to be the important part because that's where we saw the hepatopancreatic ampulla, where we saw the dumping of bile and our pancreatic juices. And it's called the duodenum. The early definition of the duodenum was 12 finger widths, hence the term duodenum. The first 10 inches are going to be the area that we'll talk about where we see continued digestion, and then we're also going to be able to see absorption. So these are the main other functions that we do need to look at. Again, reiterating the small intestine, we continue with digestion, and we start the process, we continue with the process of absorption. We saw a little bit of absorption that takes place in the mouth, a little bit of the absorption that takes place within the stomach, and then the bulk of where all the absorption takes place is the small intestine. So we've got to absorb our nutrients, we've got to absorb our vitamins, we've got to absorb our glucose as well as our amino acids, and then we'll look at the absorption of our fats, and then namely of all the things of all the secretions that we saw, we need to also absorb the biggest bulk of water. We have about a sum of 7 liters worth of water that we need to absorb, and that's all going to take place within the small intestine. This point right here that mentions the length of the small intestine, the small intestine is actually kind of scrunched up, and that's due to the smooth muscle that we'll be able to see later that holds the small intestine together. The small intestine works together. We'll see how mechanical digestion continues. Chemical digestion, we already know, are going to involve the enzymes. The mechanical digestion, again, is going to be the muscle movement that takes place within the muscularis layer. And that movement that we can see is what's called segmentations. So segmentations are the churning that takes place within each segment of the small intestine. And I'm not talking about these different segments. I'm talking about within little pieces of the small intestine. So if I could take a look at this little chunk, there are segmentations that are taking place to help mix. What I want to help illustrate is that the segmentations are not for the purposes of moving food through and moving it quickly. It's what we do to mix the stuff together. We got a bunch of chyme that's been dumped out of the stomach into the duodenum. You've got your pancreatic juices, and segmentations helps mix that stuff together. Because we've got our bile, we want to make sure that bile penetrates through the chyme. You want to get good mixing to help optimize the amount of absorption that's going to take place within the small intestine. So where this segmentation starts, it starts off right by the pylorus, and it pushes through and makes its way into each little stretch of the small intestine. So this is a reflex that takes place once that small intestine starts filling up with chyme. So what we're trying to do, again, the main idea in the small intestine is not to speed the rate at which you're pushing our chyme through. That doesn't make sense. We want to actually slow that process down. So what we saw in all of our blood vessels, right? All of our blood vessels, the arteries, the veins, we saw smooth layers of the endothelium. And we wanted that to create optimal flow so we can have uninterrupted blood flow through that vessel. We're going to see the exact opposite of that in the small intestine. Why? Because we want to slow that process down. So what you see in the mucosal layer are these circular folds, and they're called plicae circularis. These ridges are all present within the small intestine. And here you can see the jejunum actually cut up, and you can see all these little folds that chyme is going to make its way through. So you've got this, the small intestine, you've got segmentations that are coming in, and that's helping mix all of those pancreatic juices with the bile, with the chyme, and that's going to help optimize. You've got all this chyme that needs to travel through this rigid structure, or not, I'm sorry, the ridgy, that's not even a word. All these little folds that need to make its way through to encourage turbulence. And we want this turbulence to slow that process down. So with that, we have this increased surface area due to that plicae circularis. So we want increased surface area to slow this down. What it's doing is agitating that fluid as it makes its way through, that chyme that makes its way through the small intestine. Now on top of the plicae circularis, we're going to start to zoom in on each of these folds. But we can cut that plicae circularis and look at it on a tissue. Here you can see it on an organ layer. Now let's zoom in and take a look at a tissue layer where, once again, you can see the longitudinal layer. That's all responsible for the longitudinal layer of the muscularis. For the purposes of creating segmentations, we got our muscularis layer, we got our submucosal layer that'll provide the arterioles and the venules. The other thing you'll see are the lymphatic vessels that are also present, and also all the connective tissue that will hold the epithelial cells on top. And the epithelial cells on top, the structure of these are not just going to be one simple layer cell thick. In fact, what you would expect to see are what are called villi. And these villi are just the inherent structure of all these epithelial cells. They're all coming up, and they create this a bunch of little appendages that stick out on top of the plicae circularis. So here you see those folds on top. You see all these little, what they call technically they call this the brush border. And that brush border helps, once again, increase the amount of surface area. So now you've got fluid trying to make its way through. And within these villi, you'll see the structures. So within the epithelial layers, you'll see the capillary bed, and you'll see these lacteals. Lacteals are extensions of the lymphatic vessels themselves, which I'll talk about on a couple slides over. Okay? So increased surface area on the mucosal tissue. Now, if I can zoom in onto the cellular layer, now I'm zooming into one of these epithelial cells, and on top of the villi, you'll see microvilli. And microvilli are appendages on the apical side. These are going to be very similar to cilia that are right up on top, but they're not really for the purposes of moving fluid. All they do is just to increase the surface area on a cellular level. So what that does, for each columnar cell present on these villi increases that surface area quite a bit. So now that gives us more absorption that can take place. So all these combined, the microvilli, the villi present on the mucosal layer, and the plicae circularis, all increase surface area. Why do we want that increased surface area? That gives us a good interface at which the capillaries we've got a lot of capillaries right next to exposed epithelial cells that assist in the absorption. It's a good interface where we can allow the absorption of all the things that we need to absorb. Now, looking at the crevices, you can see deep within the villi, you still do get the intestinal juices. Intestinal juices are also secreted. We don't talk too much about them, but these intestinal juices basically assist. They're the same things that the gastric juices, same thing that pancreatic juices come in. These come in to assist in additional digestion. Here you saw trypsin. Trypsin comes in and allows for the breakdown of our proteins. They all get activated by stuff that's present in the intestinal juice, and that helps activate the catabolic breakdown of these proteins. There's other enzymes involved which you don't need to know, but there's other what are called colipase that help break down additional fats. Again, moving from big molecules down to small molecules once we make our way down the intestine. This just describes the microvilli. Here you can see the simple columnar cell. This is the actual electron microscope that shows what the intestinal brush border looks like. It's called a brush border because it looks like a brush. You've got your columnar cells here, and you've got all these little appendages, microvilli. These actually happen to be on top of the villi themselves. So numerous iterations of these appendages that help increase the surface area. How do we absorb? So the things that we've already broken down. We've taken our carbohydrates and break them down into glucose molecules, glucose and other monosaccharides. We have our proteins that are present in our food that get broken down to amino acids or smaller peptides. And then we had fats that were broken down into fatty acid molecules, but a good chunk of the fats were also emulsified by bile. Okay? So looking at each of these portions, you'll see that almost all of the absorption that takes place within the intestines, 90% of that takes place within the small intestine. You'll see our proteins, nucleic acids, and our sugars are absorbed into the capillaries. Either they're moving down simple diffusion, down from a high gradient, a high concentration down to a low concentration. You've got active transport to transport them against their concentration gradient, if that's the case. Our fats have a different mechanism for absorption. Remember, these are emulsified. We have to absorb these through a different passageway. How do we absorb these fats? These fats become covered in proteins. So not only are they separated by bile, but they'll get coated with proteins, and they end up being called what are chylomicrons. So chylomicrons, as I mentioned, things like your peptides and your capillaries get directly absorbed into the capillaries. Chylomicrons, on the other hand, will enter the lymphatic system. So these vessels, again, are called lacteals that project right into the villi. And these chylomicrons allow those fats to get absorbed right into the lymphatic vessels. So those lymphatic vessels will make their way in. They'll return those chylomicrons, and they'll dump that right back into the left subclavian vein. That'll make its way into the superior vena cava, back into the right atrium. So completely different pathway than that of absorption through the capillaries. Why is that the case? Actually, I don't really know why that's the case, but if you were to take a look at the content of blood, you could typically see with like with a particularly fatty diet, you'll see the presence of the fats that are absorbed through chylomicrons separately, as opposed to that the same mechanism we absorb our nutrients through the capillaries. Large intestine is going to be the last piece. So again, small intestine is absorbing a good amount of the nutrients, absorbing a good amount of all the different monomers that we've looked at. The large intestine is going to continue on with water absorption. So looking at the length of the large intestine, about 5 feet, of which you can see the ascending, the transverse, and the descending colon, and then the sigmoid colon. So 4 main parts of what we're going to be looking at of the colon itself. There's other areas that might not be overly important, or at least things that we don't really talk about too much. The cecum is actually a smaller projection. We're moving from the ileum, again, the last portion of the small intestine. You've got the cecum, which is not as developed in humans, but much more developed in herbivores. Look at cows, for instance, have a very well-developed cecum. If you look at your earlier slides, you saw species that have very well developed, and I'll talk about a little bit about that when we get to that section. we'll see the ascending colon, the transverse colon, and then the descending colon. So this is wrapping all around the cavity for the small intestine. The sigmoid colon will wrap around, and the last piece we'll talk about is rectum as well as the anal canal. Looking at the large intestine, we don't have any of the plicae circularis or the villi that you saw in the small intestine. So even though we're going to continue with some of the absorption, the thing that we typically see really is just the microvilli. So here you see the same layers, the muscularis, you'll see the longitudinal, you'll see the circular muscles within the muscularis, you'll see the submucosa again, and then the epithelial cells, the columnar epithelial cells, are going to be present in the mucosa. But right up on top, you'll see the microvilli. Again, for the purposes of absorbing. And what we're absorbing in the large intestine is mostly water. Other cells that are also present, you can see some of our goblet cells. Goblet cells that are embedded within the mucosal layer produce mucus. And that's to create, again, that thick mucus layer that's present right up at the very top. What's namely present within large intestine are what are called Teniae coli. And the Teniae coli are basically these long ribbons of smooth muscle. And what they do, and let me show you on an earlier slide, here you can see the Teniae coli that is this, again, this long ribbon of muscle. And what that does, it basically helps scrunch up. They contract to create these little different segments that you can see. So in the cases where the muscle is relaxed, you get more of a longer length of the small intestine. And when these contract, they help kind of scrunch it all up together. And why that really is the case? Why do we have that? Well, it creates what are called these small pouches called haustra, and that haustra allows the contractions to take place within each of the segments. So as the contraction takes place, it helps move the contents of the small intestine to move up through the ascending colon, through the transverse, and down the descending colon. So these are what are called haustral contractions, and they help stimulate movement of leftover stuff through the large intestine. And these haustral contractions are things that take place. Again, we'll talk about what stimulates these, but they typically take place right after eating. So if you ever eat something and you notice all of a sudden you're getting all these sort of bowel movements, what is that happening? That's not only the mixing waves that are taking place in the stomach, but also the large intestine. So it's all ramping up. Once you eat something, that's going to feed sensory information to the central nervous system and start ramping things down the alimentary canal. And this is an exact case of that. The cecum, it's a small pouch. I'm not going to talk too much about that. The colon, which are all parts of the large intestine. Again, the main purpose of these, especially the cecum, is to contain what are called beneficial bacteria. So again, this is still consistent with the external environment. We do have tons of beneficial bacteria in the large intestine that help the breakdown of stuff that we don't normally break down. Another good example of that is E. coli. We have plenty of E. coli present in the large intestine. It's something that we don't produce E. coli. E. coli is just present in there to help break down those additional nutrients. They help break them down and release the vitamins that allow us to absorb those in place. It's the sort of the spin for where probiotics come in, things that are present in yogurt. I know there's a lot of even journal articles that support fermented foods like sauerkraut, kimchi, and other fermented foods that are good for you because they house this beneficial bacteria and they help sort of replenish. If you ever hear some of those buzzwords that things like kombucha. Kombucha has tons of, even apple cider vinegar, for instance, tout that they're full of probiotics that come in and replenish the gut flora. That's exactly what they're talking about, is the presence of that bacteria in there. Now, does it really provide any benefit? I can't necessarily answer that because I know that's what we're trying to figure out. But I think the general consensus is, if you already have normal gut, if your diet is pretty normal, you already have the bacteria there. There's no sense in trying to replenish the gut bacteria because you're not flushing them out. You're not you're not lacking bacteria. You're constantly always producing it. Typically in cases that I have read about that where they may be important, when you take antibiotics kill not only only bad bacteria, they also kill a good amount of good bacteria. So antibiotics can definitely disrupt the gut flora, and that's where some probiotics can come in and help replenish that. Again, does it really help? I don't really know, but all of these things are really more homeopathic remedies that may not necessarily, they may or may not have any actual medical benefit of any kind, hence why they're considered supplements. There's certain diseases like cholera that result in extreme diarrhea. And a lot of that is flushing too. If you ever see in cases of diarrhea, you can see heavy mucus. If the mucus that's heavily present in diarrhea can contain a lot of that E. coli bacteria that's flushed out. So in responses where any disruptions of the GI tract come in, our response is pretty much to flush out whatever we're trying to get through our system. So you'll see typically things that are not digested, you get a lot of fluid that's expelled out and a lot of mucus that comes out to essentially restore and help start the process of absorption all over again. The rectum is the last portion that we'll look at for the last 5-10 slides. The rectum is the last portion of the large intestine. So now we're making our way. We know we've done a good amount of the absorption of water. We're now end up with compacted remainders of what we did not absorb. So what typically would you see is present in what's now known as feces? A lot of the stuff that we couldn't absorb. We have cellulose that's present within plant material. If you ever read on the back of food items, you'll see fiber, a lot of dietary fiber or non-soluble fibers. That's stuff that we don't necessarily absorb, but they help contribute to good diets as well as the sort of bulk of what's present in your feces. So they help with the movement of bowels. Looking at the rectum, again, the rectum is the last 8 inches. Here you can see the real last portion of the last 1 inch is called the anal canal, and that's this portion right here. And this anal canal actually have these ridges, and these ridges are these longitudinal folds that contain a bunch of, contain a bunch of arteries and veins in that area. So this area is very rich in blood supply, hence why suppositories are used. We typically, suppositories are medications that are covered in, I actually don't know what they use as a binding material, but it's typically kind of fatty. I think it's almost like coconut oil or whatever they use. Something soft, but when inserted into the anal canal, it's typically used to melt into, and then doesn't really interfere with the anal canal, but the medication can get absorbed due to that rich blood supply. I just recently watched a terrible show called, it's not really terrible, Shameless. Has anybody ever seen Shameless?
Yeah.
I think I got as far as like the end of season 7, beginning of season 8. Not to really spoil anything, but I think the father's like this really bad alcoholic, and he gets to the point where he can't really drink alcohol anymore because of all the irritation and all the whatever's happening in his esophagus, and he ends up puking quite a bit of blood. So he's just like, well, you got to get it in some way, and he tells his son to absorb alcohol the other way, and the other way was to use a funnel and a tube and to funnel alcohol through this way, and that's our way of absorption. There's a lot of drugs that utilize suppositories that help absorb this drug slowly, but that technically is the other way of doing it. It's maybe not a preferred way, but if you need to get medications in, this is your way of doing it. And certainly to kind of tie in with the guy in Shameless, this is his way of being able to get alcohol into his system. The one thing on here that I want to talk about is the internal anal sphincter as well as the external anal sphincter. So there are 2 sphincters that are present in the muscularis layer. So the internal anal sphincter is a little bit deeper. The external anal sphincter is the muscle that surrounds it. So this is called the internal, this is considered the external. So what you can see on the image to the right, the internal anal sphincter is what's going to be able to detect. There are stretch receptors that help to help give feedback if the anal canal is starting to fill up with feces. Okay, so that's going to feed back and let us know. Okay, anal canal is starting to fill up, but we still have voluntary control over defecation, and that is the external anal sphincter. This is all voluntary muscle that comes in. This is our way of basically pinching off and saying, don't release, bless you, don't release any of the feces just yet. In cases with especially babies, babies don't actually have very strong motor control. Hence why they have no control over defecation. So in those cases, the internal and external sphincter work hand in hand, or actually the external doesn't really work as well, but that in babies this causes release of feces whenever they, I guess not really feel like it, whenever babies just defecate. They defecate not at will, they just do it whenever they want to. The external anal sphincter is something that is voluntary, something that is learned that we do in order to prevent defecation. And once the stretching comes up to the point that we fill up with quite a bit of pressure in the feces, this is where the internal anal sphincter starts to pick up and sends information back, letting us know that we really need to defecate. Okay? But the external anal sphincter is what we use in order to override that. So it's a mixture of both voluntary and involuntary feedback. Now, the large intestine, just to kind of finalize the functions of the large intestine, is to think about what we're doing with all the fluids. Where does the bulk of the fluids absorbed? We mentioned 7 liters, 1.5 from saliva, 3 from gastric. We have 1.5 in pancreatic, and I'm missing one bile, that's right. One liter from bile. That comes out to 7 liters. And on top of that, the additional 2 liters that you drink, 2 liters, that comes out to about half a gallon, about 64 ounces divided by a cup. Cup has 8 ounces. That's 8 cups of water per day. So if you have heard of why do you need to drink 8 cups of water, well, that's technically how much you should be drinking per day. So about 64 ounces. So on top of that, that's about 9 liters that enter the alimentary canal. We have to absorb the remainder of that. And again, that's part of the sort of evolutionary process for us to be able to absorb our fluids. Because if we were losing that much fluid, we'd dehydrate in a matter of like a couple days. What we need to do is to look at where that absorption takes place. Now, the absorption of where that takes place is really the small intestine. The small intestine absorbs 8 out of those 9 liters. The large intestine does the final 1 liter of absorption. So that the final bit of what we have left is about 100 mils of water left over in the feces. Okay. This is our way of being able to conserve. If you look at any of the other species, especially in desert species, I know like desert rats have the ability to basically eliminate, and this is not even really covering the last portion of excretion because we still need to talk about urination. But this is our way of being able to conserve fluids. So some of the desert rats actually can bypass defecation. They can actually eliminate very hard pebbles, which will really eliminate, or at least reabsorb most of the water that takes place. And they can end up peeing out like uric crystals, so basically to really conserve on water. And that's basically what we're doing. We want to utilize that water so that we can continue reusing that to maintain normal blood pressure and maintain normal tonicity of our blood. So don't confuse that. Most people like to think that the large intestine does the bulk of water absorption. It's really the small intestine. Looking at the composition of feces, most of that, like I mentioned, is stuff that we don't absorb. But there's still a good amount of calories that are present. Calories are basically the units of energy in a certain amount of food. there's still a lot of usable energy. What we've been able to do through the process of breaking down those nutrients is we're releasing the energy contained within the chemical bonds, okay, and the state of energy of those molecules. We're able to utilize that to produce ATP. We can get glucose. From it, and that will help fuel cellular respiration. Looking at the breakdown of that food, what's left over in the feces can still be used as a viable food source. So even though feces are what's eliminated, you'll see that there's other species that can come in and still utilize the energy that's still contained within, which is all part of the sort of life cycle. But nonetheless, the rest of the other components of feces, most of that coloration, that brown coloration, comes from bile. That's leftover bile as well as bilirubin from the breakdown of our red blood cells. The odors that are present in feces, typically present from the bacteria. This is leftover bacteria. This is the gut flora that's present to help the breakdown of our food. Some of these cells, this particular slide goes into the other beneficial bacteria. We have tons of different microorganisms. And the bulk of our feces really is those leftover cells, or leftover microorganisms. Like I said, the presence of those bacteria help the breakdown of leftover food. You can see vitamin K is something that's also important, biotin, other amino acids that are all absorbed due to that beneficial bacteria that help break this stuff down. The last couple things I want to talk about are the reflexes that are involved. The reflexes that are involved that help govern the motions of the large intestine. So now that we know how the large intestine helps create these haustral contractions, let's talk about what triggers it. So the things that we talked about earlier also play a major role. You'll see the two autonomic reflexes are the gastroileal reflexes as well as the gastric colic reflexes. The biggest bulk of what's going to create these haustral contractions is gastric filling. Again, looking at the chemoreceptors located in the stomach, looking at the stretch receptors in the stomach. Once food enters the stomach, that starts the process of all these peristaltic movements, whether they're mixing waves, whether they are whether the release of hormones, our cholecystokinin, our gastrin, the release of pancreatic juices, release of bile, causing our segmentations in the small intestine as well as the haustral contractions taking place in the large intestine. All of these reflexes come into play to help move and propel that stuff down the alimentary canal. The big ones, though, are the gastroileal reflex and gastrocolic reflexes, and our next slide talks about those. The gastroileal reflexes is just peristalsis in the ileum that causes relaxation of the ileocecal valve. Not something I'll really ask you, but basically what all it does is it pushes stuff from the small intestine, the last portion, the ileum, pushes it right into the cecum. So this is our first bit, when we move from small intestine to large intestine, that causes large haustral contractions in the large intestine. The other one is a gastrocolic reflex, again, promoting a lot of muscle movement within the transverse colon. So you get these pretty much, where is it taking place? Pretty much within the transverse colon. So as gastroileal reflex takes place right here, when the stuff is present within the transverse colon, you'll get a lot of contractions that are taking place, and that is just right up above. If you ever eat something and you feel the sort of bowel movements, listen to anybody. If you ever okay, never mind, I won't mention that. But really all I was trying to get into is like if you ever hear somebody eat, or if you know somebody eats, listen to their stomach, and then I realized that's really creepy. Don't do that. But a lot of the motions you can hear that are taking place, people like to think it's the stomach. It's actually not the stomach. This is the transverse colon. Again, the gastrocolic reflex that's taking place due to the presence of food in the stomach. That is our way of ramping up to get the small intestine to start moving. And again, they work all through the 10 E. coli that help promote those haustral contractions. These actually take place quite a bit, and when do those reflexes take place? Right after a meal. So they, just again, due to the presence of a meal, once food enters the stomach, starts triggering these contractions. And those contractions that are taking place can start increasing the pressure within the large intestine, start increasing the amount of pressure in the anal canal, which again triggers feedback into the involuntary sphincter, giving us the feeling of the need to defecate. So if you know of anybody, and I certainly know a couple people that once they start eating, immediately they feel they need to go to the bathroom. Why is that? Haustral contractions encourages that. The defecation reflex is basically everything that I've talked about that results in the process of defecation, again, the elimination of feces. Food in the stomach stimulates peristalsis, we already described. Moving food through the intestine into the rectum. The presoreceptors, again, these are pressure receptors. They're like baroreceptors, but they actually just detect pressure. So as the feces presses up against these rectal presoreceptors, that causes distension as well as the longitudinal muscles. And I'll show you back in our prior slide of how that takes place. So there's longitudinal muscles, again, that I mentioned that are on the outside of the muscularis. What happens as the presoreceptors come in and start filling up with feces, this starts pressing against these presoreceptors that ends up shortening the length of the rectum. So the longitudinal muscle starts to scrunch up altogether. And that actually causes positive feedback because that presses up against the internal anal sphincter. So as this starts to fill up, contraction of the longitudinal muscle, increased pressure, positive feedback. Increased pressure then sends feedback information letting you know that you do have that urge to defecate. And again, all that defecation is all managed through contraction of the external anal sphincter. And then, of course, with the release of that external anal sphincter is where defecation actually takes place. And that's how we end up eliminating all of our wastes. With that, I've got nothing left. That last slide is something I've been meaning to get rid of, and I don't know why that's on there, but colonoscopy, I was just looking at what they would do. I think what I really wanted to do was just illustrate what the look would look like in the large intestine. You can see the different segments that are present within, and again, that's all due to the 10 coli that help contract. So just know for a colonoscopy, they do tell you not to eat anything, otherwise it looks like that to the right. All of this helps summarize. This entire table goes in and summarizes all of the different organs. So